Hello champs, I hope you all are doing great. My name is Biswajit, I'm your botany teacher. Today we are going to discuss a very important, conceptual and interesting chapter of class 12th, and the chapter is molecular basis of inheritance. That means in this chapter we are going to discuss which type of molecules and how the molecules are responsible for transmission or the transfer of genetically based characters from one generation to another generation. And this lecture or this chapter requires your honest attempt or honesty, I would say. I have seen many students, they face problems to have proper understanding of various concepts like operon concept, the post-transcriptional modification process, DNA replication, DNA fingerprinting, etc. etc. But I would say, I would assure you that if you watch this lecture honestly, you won't face any problem to have proper understanding of those chapters. So the topics which we are going to cover today are DNA, search for genetic material, RNA, RNA world, DNA replication, transcription, post-transcriptional modification, translation, gene regulation, genetic code, central dogma, and apart from this chapter also includes DNA fingerprinting and human genome project. And I will discuss those topics in the next chapter or in the next lecture along with ecosystem. But in this class, I will explain you the terms or the techniques which we use in DNA fingerprinting. Okay, so don't think that I'm skipping those portions. I will explain you, but I will explain only the terms or the techniques which we use in the DNA fingerprinting. In the next class, I will explain the mechanism of DNA fingerprinting. Why? Because it requires a lot of time to understand those topics. Okay. So when I talk about the organisms, every organism has characters and the characters are controlled by genes, and the chemical or the biochemical which contains the gene are called as genetic material, and the genetic material is always nucleic acid. Whether the organism is non-cellular, for example virus or viroid, or cellular like prokaryotic or eukaryotic organisms, every organism has genetic material. And the genetic material is always nucleic acid. Yes, the genetic material can be DNA or can be RNA depending upon the nature of organism. If the organism is cellular organism, then the genetic material is always DNA and the DNA will be double-stranded DNA. In case of prokaryote, the DNA is circular in nature except mycoplasma. But in case of eukaryote, the DNA is linear. I repeat, every cellular organism has double-stranded DNA as genetic material. And if I talk about the prokaryotic cells, the DNA will be circular in nature. And in case of eukaryotic cells, and especially in the nucleus, the DNA are linear in nature. Okay, but there is one exception. In mycoplasma, even though this is a prokaryote, it has linear DNA. Okay, so in these organisms which have these cells, the DNA acts as genetic material. It does not mean that only DNA is present in those cells. No. Along with the DNA, RNA are also present. But the RNA molecules like mRNA, rRNA, tRNA, snRNA or snRNA, etc., they are present along with DNA, but those RNA, they do not act as genetic material. So if any structure, DNA and RNA both are present in any cell, DNA and RNA both are present, then only the DNA is going to act as genetic material. Clear? So we are going to talk about that genetic material, that is the DNA, which controls various types of characters. Okay, and the DNA is basically a type of nucleic acid, right? So the nucleic acid is a polymeric compound. It is a polymeric compound and it is a polymer of nucleotides. And the nucleotide, whether the nucleotide is ribonucleotide or deoxyribonucleotide, every type of nucleotide consists of three components. What are those? One is sugar. The sugar can be ribose or can be deoxyribose. Okay, accordingly we classify the nucleotide as ribonucleotide or deoxyribonucleotide. And the second component is nitrogenous base, and the nitrogenous base are of two types. One is purine, second is pyrimidine. We will talk about them in detail later on. And these two components, sugar and nitrogenous base, when they combine together with the help of beta and glycosidic bond, they form a complex, and that complex is known as nucleoside. So nucleotide consists of three components and nucleoside consists of these two components. And when I add the phosphate group or phosphoric acid to this nucleoside, that is the sugar and nitrogenous base, I will get the nucleotide. And that is why I can say that nucleotide is nothing but the nucleoside plus phosphate. And in a nucleotide, the number of phosphate can be 1 or 2 or 3. Depending on that, we classify nucleotide into as nucleoside monophosphate, nucleoside diphosphate, or nucleoside triphosphate. Clear? So if I write NMP, that means nucleoside monophosphate. And when I say nucleoside monophosphate, that means in that nucleoside the sugar is ribose sugar. Okay, but if I write DNMP, that means deoxynucleoside monophosphate. That means in this case the sugar will be deoxyribose sugar. Clear? And if I write NDP, that means this is nucleoside diphosphate in which the sugar is ribose sugar. And if I write DNDP, that means deoxynucleoside diphosphate. And if I write NTP, that means this is nucleoside triphosphate which has 3 phosphate groups. Okay, and in this case the nucleoside has or nucleotide has ribose sugar. And if I write DNTP, that means it will have the deoxyribose sugar. Clear? So I can write NMP or DNMP, okay, or NDP or DNTP, or I can write NTP or DNTPs. These are the examples of what? Nucleotides. Clear? So we are going to discuss all these components one by one. So whether the nucleotide is of RNA or DNA, that means the whatever the nucleic acid is, the sugar is always pentose sugar. That means the sugar will have 5 carbons. So because the sugars which are present in the nucleotide, they have 5 carbons, that is why they are called as pentose sugar. And these are those pentose sugars. Clear? And these pentose sugars, they are cyclic structures, and in that cyclic structure oxygen and carbon are present. So here I am having oxygen, and apart from oxygen, carbon will also present. Carbon are also present. Okay, so in this ring structure I have oxygen as well as carbon, and that is why I call these ring structures or the sugars as heterocyclic structure. Hetero, why? Because the ring consists of carbon and oxygen. Clear? Here also I will have carbon, here also I will have carbon. So this is ribose sugar. So this structure is also ribose sugar. But if I remove this oxygen from this OH group, so if I am removing oxygen, that means I will call deoxy. So from this structure, if I remove the oxygen, then I will have this hydrogen, and that is why I will call this structure as deoxyribose. So what is the difference? The difference is, in this case I have OH group, hydroxyl group, but in this case I have hydrogen. So what about the numbering of these carbons? Okay, so the carbons are numbered like this. So here the carbon is numbered as 1 prime, this is 2 prime carbon, this is 3 prime carbon, this is 4 prime carbon, and this is 5 prime carbon. Okay, so here also this will be 1 prime carbon, 2 prime carbon, 3 prime carbon, 4 prime carbon, and this is 5 prime carbon. Clear? So the difference between ribose and deoxyribose sugar lies at the 2 prime carbon. In this case, the 2 prime carbon has OH group. It has OH group, right? So it has, just a minute, it has OH group, whereas in this case the 2 prime carbon has hydrogen. This is the difference. Okay, and this ribose sugar is present in the RNA, in the ribonucleotide, whereas this deoxyribose is present in DNA. That means I can say this is present in the deoxyribonucleotide. Okay, and because these sugars, they have OH group, That is why they will interact with water, and that is why they are hydrophilic in nature. I can say because of the presence of OH group, they are polar in nature. Okay, and I have already mentioned that the sugars present in the nucleotide, I'm not saying about sugar as normally, okay, in general. I'm not saying about sugars in general. I'm talking about the sugars present in the nucleotide. They are either either ribose sugar or deoxyribose sugar. Okay, now the second component. Now the second component about of this nucleotide is nitrogenous base. Okay, because this compound or the component has nitrogen, that is why we call this as nitrogenous base. And this structure is also heterocyclic. Why this is heterocyclic? Because it has carbon and nitrogen. It has carbon and nitrogen. In case of sugar, why was that heterocyclic? Because that was having carbon and oxygen. In this case, it is having carbon and nitrogen. Okay, and these nitrogenous bases, they are non-polar. They are not going to interact with water. They will stay away from the water, and that is why they are hydrophobic. This point is important. This nitrogenous base is hydrophobic, whereas the sugar is hydrophilic. Okay, and this nitrogenous base which is present in a nucleotide can be purine or pyrimidine, and the purine is of two types in nucleotide. One is adenine, another is guanine, and this purine has double ring structure. It has a double ring structure, and that double ring structure contains 9 members. It contains 9 members. Okay, and in the pyrimidine, it has a single ring structure and it has 6 members. Purine is double member ring structure. Pyrimidine is single member ring structure. Purine has 9 members in its ring, whereas pyrimidine has 6 members in its ring. And the examples are cytosine, thymine, and uracil. Cytosine and thymine, they are present in DNA, whereas thymine is present in DNA, whereas uracil is present in RNA. Uracil is present in RNA. But there is one exception, and the exception is tRNA, transfer RNA. And in transfer RNA, there is a loop which is called as T-Cy loop, and the T-Cy loop has T, thymine. Otherwise, if I say in general, I would say the thymine is absent in RNA, and it is present in DNA, and the uracil is present in the RNA, and it is absent in the DNA. Okay, so what about cytosine and adenine guanine? They are present in both DNA as well as RNA. Okay, now what about the phosphate in a nucleotide? The phosphate, they are associated with the sugar molecule. They are associated with the sugar molecule. So let me write that part. Okay, so they are associated with sugar molecule. Okay, so sugar has 5 carbons. So which carbon is of the sugar is associated with this phosphate? It is the 5' carbon. It is the 5' carbon. It will be associated with the phosphate group, and the linkage is phosphoester linkage. The linkage is phosphoester linkage between the 5' carbon of the sugar and the phosphate. Okay, so let me explain the various types of linkages in a NTP, nucleoside triphosphate. So this is a nucleoside triphosphate. This is nitrogenous base, this is sugar, and these are the phosphates. So phosphate plus sugar plus nitrogenous base, that will be nucleoside triphosphate. Okay, so the linkage which is present between this nitrogenous base and this sugar, this is sugar and this is nitrogenous base, and here I will have this 1' carbon. So this is 1' carbon, this is 1' carbon, and this is 5' carbon, right? This is 2' carbon, this is 3' carbon, this is 4' carbon. Okay, so I can write here carbon, 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 like this. So the 1' carbon of this sugar is associated with the nitrogen of this nitrogenous base. Okay, and the linkage will be beta and glycosidic linkage. Which linkage? Beta and glycosidic linkage. Beta and glycosidic linkage. Okay, the first point you have to remember here is the linkage between sugar and nitrogenous base is beta and glycosidic linkage. Okay, and the member of this nitrogenous base which is associated or which is involved in the formation of beta and glycosidic linkage is 1 number nitrogen or 1 number member of this nitrogenous base or 9 number member of the nitrogenous base. So let me write that here. Okay, so who are associated over here? So it is the 1' carbon of sugar, okay, and 1 first nitrogen of pyrimidine or 9th number nitrogen of purine. If this nitrogenous base is purine, then at the 9th number there will be nitrogen, and that nitrogen will be involved in the formation of this bond. And if this nitrogenous base is pyrimidine, then the first nitrogen will be associated with the formation of this beta and glycosidic bond. Clear? And here I am having phosphate. So this is phosphate and this is sugar. And the linkage here is the phosphoester linkage. This is phosphoester linkage. Why this is phosphoester linkage? We know very well the ester linkage is C double bond O oxygen. This is ester linkage. And if I write P instead of carbon, it will be phosphoester. It will be phosphoester. And you can clearly see here, this is phosphoester linkage. That is why I will say the sugar and the first phosphate, they are linked by phosphoester linkage. Clear? And if you are able to understand, if you are understanding this, please do mention in the comment box. That will encourage me. Okay? And remember one thing, the phosphates which are present in this nucleoside triphosphate, they are named as alpha phosphate, beta phosphate and gamma phosphate. Okay? And here I am having, so what is the linkage present between the alpha phosphate and beta phosphate? It is the phosphoanhydride linkage. Which linkage? Phosphoanhydride linkage. So this is phosphoanhydride linkage. This is not phosphodiester. No. Many students, they make this mistake, they commit this mistake that they would say, okay, the linkage is phosphodiester. No, it is the phosphoanhydride. The linkage between two nucleotides is phosphodiesterlinkage. Here I am talking about the bond between the two phosphate groups, not two nucleotides. So this is phosphoanhydride. So what is the anhydride? All of you know very well. If I write like this, this is anhydride linkage. And instead of carbon, if I write P, this will be phosphoanhydride linkage. This will be phosphoanhydride linkage. And the same thing is present here. You can see this quite clearly, right? So here you can see this is phosphoanhydride linkage. This is phosphoanhydride linkage. Clear? Now, here also I am having the same linkage. Here also I am having the same linkage. So these linkages are phosphoanhydride linkage. Okay, that means sugar and first phosphate, phosphoester, alpha beta phosphate, phosphoanhydride linkage, and beta gamma phosphate, it is phosphoanhydride linkage. So these are the various linkages present in a nucleoside triphosphate. Okay. Now let us talk about the structure of DNA. The DNA present in a cell, or the DNA in general, can exist in various forms, like A form of DNA, B form of DNA, Z form of DNA, etc., etc. And the most common form of DNA is B form of DNA. Which DNA? B form of DNA. And Watson and Crick, sir, they gave the double helical model of the structure of DNA of bDNA, not aDNA or zDNA. Okay, so here whatever the points we are going to discuss here are with respect to bDNA. Which type of DNA? bDNA. The most common form of DNA, that is the bDNA. Okay, so the double helical structure of bDNA was given by Watson and Crick, sir, and they took the help of the X-ray diffraction pattern which was obtained by Franklin and Wilkins, sir, and they also used the Chargaff rule to explain the structure of the DNA. Okay, so first of all, let us talk about the Chargaff rule. Okay, then we will talk about the salient features of the bDNA, double helical structure of bDNA. Clear? So when I talk about the Chargaff rule, it was given by Chargaff, and this rule is applicable to DNA only. But DNA exists in both single-stranded form and double-stranded form. So do you think that this rule is applicable to single-strand DNA? No, this is applicable only to double-strand DNA. This rule can also be applicable to double-stranded RNA. But here we will talk only with respect to double-stranded DNA, not single-stranded DNA. Okay, so this rule is applicable to double-stranded DNA. This is very, very important. This is applicable to double-stranded DNA, not single-stranded DNA. Okay, and this was given by Chargaff. And what does this rule say? This rule says that in a double-stranded DNA, the number of purine will always be equal to the number of pyrimidine. Okay, and we know what are the purines in a DNA? The purines are adenine and guanine. What about the pyrimidine? The pyrimidine in DNA are cytosine and thymine. So I can say that A plus G will always be equals to C plus T, and this is true in case of double-stranded DNA. And that is why I can also say that A plus G upon C plus T will be 1, and this ratio is always 1. This ratio is always 1. Okay, and in a double-stranded DNA, the nitrogenous bases are present 100%. Clear? So if A plus G plus C plus T, they are 100%, then I will say that A plus G, okay, so A plus G plus C plus T, 100% nitrogenous bases, 100% nitrogenous bases, and because A plus G is equals to C plus T, that is why I can say here that A plus G will be 50% and C plus T will be 50%. In a double-stranded DNA, if I consider that the sum total of nitrogenous base is 100%, then I will say A plus G will be 50% and C plus T will be 50%. Why? Because A plus G is always equals to C plus T. A plus G is always equals to C plus T, and that is why if I say suppose in a double-stranded DNA, let me write that question. So if in a double-stranded DNA, the percentage of A is 30%, A is present 30%. So what will be the amount of cytosine? What will be the amount of cytosine? Clear? So here I will say I know that adenine plus guanine is 50%. Adenine plus guanine is 50%. So if adenine plus guanine is 50%, then I will say that is why in this case G will be 20%, because the A is present 30%. And Chargaff, he said that the amount of adenine present in a double-stranded DNA will always be equal to the amount of thymine, and the number of or the amount of guanine in a double-stranded DNA will always be equal to the number or the amount of cytosine present in a double-stranded DNA. So I will say if the amount of G is 20%, that is why the amount of cytosine will also be 20%. It will be 20%. Clear? Great. And then he said that A plus G upon C plus T is always 1. This ratio is always 1. Whether the DNA is from prokaryote or eukaryote, whatever the source is, it does not matter, but this ratio is always going to be 1. But if I talk about A plus T upon G plus C, this ratio is not 1. This ratio is not 1. Okay, so what will be the value of this ratio? The value of this ratio can be more than 1 or can be less than 1. If this ratio is more than 1, that means if this ratio is more than 1, that means in the DNA A and T they will be present more as compared to G and C. And that is why I will say that the DNA will be AT rich DNA. And where do you find such DNA? In case of eukaryotic cells. In case of eukaryotic cells. So that is why I will say in case of eukaryotes, the DNA is AT rich. The DNA is AT rich. And if the amount of G and C is more as compared to A and T, in that case this ratio will be less than 1. And they are found in the prokaryote. So I will call those DNAs GC rich DNA. I will call those DNAs GC rich DNA. And remember one thing, the organellar DNA like mitochondria and plastid DNA, they are also GC rich. They are also GC rich. Okay, so here I will write here one more point. Okay, so I will write here mitochondrial and plastidial DNA, they are also GC rich. They are also GC rich. Okay, so these are the various points of Chargaff's rule. So the Chargaff's rule is always applicable to double strand DNA. Okay, so if I give you one data, and if that data says that suppose in the double strand DNA the amount of adenine is 20%, okay, and thymine is 10%, okay, and guanine is suppose 30%, like this, and suppose cytosine is present 40%. So from this we can conclude that this DNA is not double stranded. Why not double stranded? Because in a double strand DNA the amount of adenine will always be equal to amount of thymine, but in this case adenine and thymine, they are present in different amount, right? So from this data we can say that DNA is single stranded, not double stranded. Okay, so here I will say this DNA is single stranded, not double stranded. Okay, now so here I am having one question. I will give you one question here. So suppose there is a DNA which has 1000 base pair. BP is base pair. We can define the length of DNA in terms of base pairs or nucleotide pairs. Okay, so suppose in the DNA adenine is present 30%. 30% adenine, 30% of the nitrogenous bases are adenine. So what will be the total hydrogen bonds in that DNA? So here we should know one thing, that the hydrogen bonds present between adenine and thymine is 2 and guanine and cytosine is 3. This is first thing which we should know. Second thing, it says that the total nitrogenous bases, or what is the size of DNA? The size of DNA is 1000 base pair. So if 1000 base pair, that means what will be the total number of bases? It will be 2000 bases. 2000 bases are there. So what is amount of A? It is 20%. It is 20%. So 20% adenine, that means it will be 2000 into 20%. That means 20 into 100. This will be the number of the adenine. This will be the number of adenine. Clear? So it will be 400. It will be 400 adenine. So if adenine is 400, so there will be thymine will also be present 400. 400 thymine will be present. Both adenine and thymine, they are present 400. And what about guanine then? We know adenine plus guanine is 50%. And because adenine is present 30%, that is why the guanine will be present 20%. Sorry, here I have to write 30%, right? here, suppose I can change this question, no problem. I will change this question. Suppose here it is 20%. Now this is clear. Okay. So in this case, the guanine will be 30%. The guanine will be 30%. Right. So if guanine is 30%, that means it will be 30% of what? 2000 bases. 30% of 2000 bases. So I will say, that is why I will say guanine will be 2000 bases 30%. Right. So it will be 600. It will be 600. So because guanine is present 600, so that is why number of cytosine will be also be 600. And we know adenine pairs with thymine and guanine pairs with cytosine. Okay, so I will have 480 base pair. 80 base pair will be present 400, and GC base pair will be present 600. 600 GC base pair are present. So what will be the total hydrogen bonds? So here the total hydrogen bonds will be 400 into 2, because between A and T 2 hydrogen bonds are present, and between G and C I have 3 hydrogen bonds, so it will be 600 into 3. So it will be 600 into 3. So it will be 800 plus 1800. So it will be 2600 hydrogen bonds. The hydrogen bonds will be 2600s. Okay, so this should be your approach to solve the questions. Very easy. Now let us talk about the salient features of the double helical DNA. Double helical DNA, which was given by Watson and Crick. So Watson and Crick, sir, they said that the DNA, a double-stranded DNA, will consist of 2 single-stranded DNA, 2 chains of single-stranded DNA. Okay, so here this is one strand of the DNA, and this is one strand of DNA, and this one is another strand of DNA. Okay, and those 2 strands, they are coiled in a right-handed manner. They are coiled in a right-handed manner. That means, suppose these are 2 strands. These are 2 strands. Okay, and before explaining that, let me tell you one more point. Suppose these are 2 strands, and these 2 strands are anti-parallel to each other, and that is why the DNA is anti-parallel. What do you understand by this? It means the 2 strands, they have opposite polarity. And when I say opposite polarity, that means in this strand, at this end phosphate will be present, at this end OH will be present, and the phosphate which is present, the end which has the phosphate group, that will be called as 5' end, and the end which has OH group will be called as 3' end. It will be called as 3' end. So this is 3' end, and this will have phosphate, so this will be 5' end, and this will have the OH group, so this will be 3' end. So you can clearly see the polarity is 5' to 3', and here 3' to 5'. So they are anti-parallel. They are anti-parallel. So this model given by Watson and Crick, sir, that says that a double-strand DNA consists of 2 strands, and each strand one strand has 5' to 3' polarity, whereas other strand has 3' to 5' polarity. That is why they are anti-parallel. And each strand consists of a backbone. It consists of a backbone, and that backbone is made of alternate sugar and phosphate. Alternate sugar and phosphate. Okay, and you can clearly see from here. Here you can clearly see that this is a nucleotide chain. This one is nucleotide chain. Okay, and here in this case, this one is the backbone of this nucleotide chain. Okay, so this one is the nucleotide chain. That means this is the single-stranded DNA. Okay, and this one is also single-stranded DNA. Clear? And this one is called as the backbone of this single-stranded DNA. And the backbone is made of what? Phosphate and sugar. Phosphate, sugar, alternate sugar, phosphate. Alternate sugar and phosphate. And the end which has the phosphate group, that will be called as 5' end. That will be called as 5' end. Why? Let me explain you here. You can clearly see that which atom of the sugar has phosphate? It is the 5' carbon. It is the 5' carbon. And the 3' carbon has OH group. And that is why I will say whenever phosphate is present, that end will be 5', and whenever OH group is present, that will be 3' end. Okay. So this one is the backbone, and this backbone is made up of what? Sugar and phosphate, and this sugar and phosphate both are hydrophilic, and because they are hydrophilic, that is why they will interact with the water. So in this end, what will be present? Water will be present. So here water will be present, and this water molecule will interact with this phosphate as well as sugar. Why? Because they are hydrophilic in nature. Okay, so here also I am having this backbone of this single strand DNA, and the backbone is again made up of what? Again made up of alternate sugar and phosphate. So in this case, this will be 5' and this will be 3'. Okay, and you can clearly see that on the inner side of this double strand DNA, this anti-DNA is double strand DNA. On the inner side of this double strand DNA, I have nitrogenous bases. Okay, so here also there will be water. Here also water will be there. So this water will interact with sugar as well as phosphate. But being hydrophobic, this nitrogenous base, they are present on the inner side of this DNA. And because they are present on the inner side of this DNA, in this case, on the inner side, there is no water. There is no water. That is why they are protected from the aqueous environment. They are protected from the aqueous environment. This nitrogenous base is not present on the outer side. Why? Because on this outer side, water is present. And this nitrogenous base is hydrophobic, so it has to minimize its contact with water. That is why this nitrogenous base has to be present, or must be present, on the inner side rather than the outer side. Clear? Great. So if you are able to understand, if you are understanding, please leave comment. Okay, that will encourage me a lot. Thank you. So here I will say the DNA, double strand DNA, consists of 2 strands. This is one strand and this is another strand. Each strand has backbone, and the backbone is made up of alternate sugar phosphate. So here P is not phosphorus, P is phosphate. Okay, so alternate sugar and phosphate are present. Okay, and to the sugar molecule, nitrogenous base is attached. Okay, and suppose here the nitrogenous base is adenine. So here it will be thymine. That means because Watson and Crick, sir, they use Chargaff's rule, and Chargaff's rule says that, everyone knows this very well, that adenine always pairs with thymine and guanine always pairs with cytosine. So here, what will be the number of hydrogen bonds? The number of hydrogen bonds will be 2 in nature. So these are 2 hydrogen bonds. 2 hydrogen bonds. Clear? Suppose in this case, suppose the nitrogenous base is guanine. So here the nitrogenous base will be cytosine. Okay, and in this case the number of hydrogen bond will be 3. How many hydrogen bond will be present? 3 hydrogen bond will be present. And suppose here I am having cytosine. So in this case guanine will be present, and here also the number of hydrogen bond will be 3. Number of hydrogen bond will be 3. Okay, and in this case I will say cytosine is complementary to guanine, and guanine is complementary to cytosine, adenine is complementary to thymine, and thymine is complementary to adenine. Okay, fine. And this model says that in a double strand DNA, purine always pairs with pyrimidine. Purine always pairs with pyrimidine. It is you will never find that in a double strand DNA, purine will pair with purine, or pyrimidine will pair with pyrimidine. No. Purine will pair with pyrimidine only. And because purine always pairs with pyrimidine, and pyrimidine they never pair, or purine they never pair, that is why the width of the DNA will always be same. The width of DNA will always be same. Okay, so what is responsible for uniform width of DNA? It is the pairing of the nitrogenous bases. And which type of pairing? Purine will always pair with pyrimidine. Purine will never pair with purine, or pyrimidine will never pair with pyrimidine. Let me explain you that part. When I started this lecture, I told you about nitrogenous bases. Purine is double ring structure. Okay, so suppose these are 2 rings of the purine, and suppose these are another 2 rings of the purine. If this is adenine and if this is guanine, okay, so they will pair with each other, but they in reality they do not pair. Okay, I'm trying to explain this, a hypothetical condition. And suppose I'm having pyrimidine, and suppose this is thymine, and suppose this is adenine. So suppose this is cytosine. and here the pyrimidine are pairing with each other. And suppose in this case one purine is pairing with, and suppose this is A and suppose this is T. They are pairing with each other. So you can clearly see that at this point the width is this much, the width of DNA is this much. At this point the width of DNA is this much, and at this point the width is little more. And this type of pairing never takes place. Only this type of pairing will take place. Okay, so suppose, let me write here one more example. Suppose I'm having adenine, or suppose let me write here, suppose guanine is present. So here I will have the cytosine. So width will be same width will be present. So here I will say purine pairs with pyrimidine, Okay, so because in a double-strand DNA, it is the purine is always going to pair with pyrimidine. That is why the width of DNA is always uniform. Okay, and the DNA which is present in a cell, they are right-handedly coiled. Okay, and when I talk about this DNA, when they are coiled with each other, it will form some grooves, and the grooves are of two types. One is minor groove. So this one is called as minor groove, and this one is called as major groove. Okay, so this one is major groove and this one is minor groove. Okay, and when I coil the DNA 360 degree, so in that one turn or complete turn, what do you understand by complete turn? That means when I turn it 360 degree. So when I turn the DNA in 360 degree, the number of nitrogenous bases or base pairs present will be 10. It will be 10. So this is one complete turn. This is one complete turn of DNA. Okay, so this complete turn consists of how many base pairs? 10 base pairs. 10 base pairs will be present. Okay, and what is the pitch of the DNA? The pitch of the DNA is 3.4 nanometer, or I can say 34 angstrom. So what do you understand by pitch of DNA? Pitch of DNA means the rise of the helix per one complete turn of DNA. Okay, so this distance, so this is one complete turn, this is one complete turn. So in a complete turn, what is the height of the DNA? What is the rise of the helix? It is 3.4 nanometer. Okay, and what is the distance between two base pairs? It is 3.4 angstrom. Okay, so the distance between base pairs is 3.4 angstrom, or I can say 0.34 nanometer. Okay, so in a pitch, what is the number of base pairs? 10 base pairs, and that is why I will say the distance will be 3.4 nanometer, or I can say 34 angstrom. Clear? And what are the forces? Which are responsible for? Okay, so here those points I have mentioned here. Okay, the DNA consists of two polynucleated chains. They are antiparallel to each other. The backbone is consists of alternate sugar and phosphate. They are hydrophilic. That is why they will interact with the external water. They are complementary to each other. Okay, and the nitrogenous bases are present on the inner side, and they are protected from the outer water. One chain has 5' to 3' polarity, whereas the other chain will have 3' to 5' polarity. And in a double-strand DNA, adenine always pairs with thymine and guanine always pairs with cytosine. Okay, and that is why I can say purine always pairs with pyrimidine. And because of this, the width of DNA, which DNA? Double-strand DNA, is always uniform. It is always uniform. And what is the width of the DNA? The width of the DNA is 20 angstrom. It is 20 angstrom. Okay, that means in this case, in this case, this width is 20 angstrom. This is 20 angstrom. Okay, now, and the two nucleotides of the same chain, of the same polynucleotide chain, they are held together, or they are linked by phosphodiester linkage, and the phosphodiester linkage is 3' to 5' phosphodiester linkage. 3' to 5' phosphodiester linkage. Okay, now what are the forces which are responsible for stabilizing the double helical structure of DNA? There are two main forces. One is hydrogen bond, second is stacking force. So this hydrogen bond, which are responsible for stabilizing the double-stranded DNA, they are present between the nitrogenous bases. For nitrogenous bases, I will write NB. Okay, and the nitrogenous bases, the hydrogen bonds are present between the nitrogenous bases of two opposite strands, complementary strands. So I will write nitrogenous bases of complementary strands. Which strands? Complementary strands. Okay, what about the second force? The second force which is responsible for stabilizing the double helical structure of DNA, it is the stacking force. And what do you understand by this? Suppose in a double-stranded DNA, so suppose in this diagram you can see that this is one base pair, this is one base and this is another base. So I will say this is one base pair and this is another base pair, I will say. So in between these two base pairs, there will be some force of interaction, and that force of interaction between the two base pairs, suppose this is one base and this is one base. So this is one base pair, this is another base pair. So in between these two base pairs, the force which is present is called, or the force of interaction between these two base pairs is stacking force. And the main force which is responsible for stabilizing the DNA, it is stacking force, not the hydrogen bond. Very, very important. Many students or many persons, they have this misconception that the main force responsible for stabilizing the structure of DNA is hydrogen bond. No, that is incorrect. It is the main force is stacking force. Hydrogen bond is also responsible, but main force, when I talk about main force, it is the stacking force. Okay, so I will say, what is stacking force? The force of interaction. It is the force of interaction which lies between two base pairs, two adjacent base pairs. Okay, two adjacent base pairs. And I can also say many points, but here I'm not explaining those points. Okay, and if you want to learn more about stacking force and hydrogen bonds, you can go and watch the lecture of biomolecule. Okay, there I have mentioned that part, more about the stacking force and hydrogen bonds. Okay, standard hydrogen bond, non-standard hydrogen bond, what are those? Okay, and what do you understand by stacking force is sequence dependent. Those points I have mentioned there. Clear now? But they are not important for NEET exam. So this is all about the DNA molecule. And when I talk about the DNA molecule, in a DNA molecule, I have phosphodiester bond between the two nucleotide pairs, two nucleotides. So what will be the total number of phosphodiester bond in a DNA molecule or RNA molecule? Okay, so here that is what I'm going to write here. Number of phosphodiester bonds in a single stranded nucleic acid. This nucleic acid can be RNA or DNA, and double stranded nucleic acid. This nucleic acid can be DNA or RNA. So if the nucleic acid, whether it is DNA or RNA, is linear in nature, in that case, the number of phosphodiester bonds will be n-1. And if the DNA or RNA, single-strand DNA or RNA are circular in nature, the number of phosphodiester bonds will be n. So here, what is n? In this case, n is number of bases or base pairs. Okay, when I will say bases, if the nucleic acid is single-stranded nucleic acid. For single-stranded nucleic acid, I will use bases. For double-stranded nucleic acid, I will use base pairs. I will use base pairs. Okay, and if the nucleic acid is double stranded and linear, in that case, what will be the what will be the total number of hydrogen phosphorus bond? It will be 2n minus 2, 2n minus 2, 2n minus 2. Okay, and here if it is circular, it will be 2n, it will be 2n, it will be 2n. Okay, so let me write one thing here. In this case, just a minute. So suppose one DNA has 3 base pairs, and one DNA has 3 bases. So in this case, if the DNA has 3 bases, so what will be the phosphodiester bond? Let me write that part. So suppose these are the nucleotides, and these will be phosphodiester bond. So this is one phosphodiester bond, this is another phosphodiester bond. PD is phosphodiester bond. Okay, and if the DNA is double stranded, so in that case, 2 chains will be present. So in this case, these are phosphodiester bonds. Okay, so in this case, what is the number of phosphodiester bond? It is 2. That means I will say n minus 1. So here I am putting the formula of n minus 1, where n is number of bases. Okay, so in this case n is 3, so the answer will be 3 minus 1, so it is 2. In this case, 3 base pairs are there. Okay, so if 3 base pairs are there, so the answer, what is the phosphodiester bond? 4 in nature, so it will be 2 into 2n minus 1. So 2n minus 2, so it is 2n minus 2, so it will be 2 into base pair. What is the number of base pair? It is 3, so it will be 3 minus 2, so it will be 4. It will be 4. Okay, so this is about linear DNA. So these DNA are linear in nature. What about circular DNA then? So suppose I am having DNA which are circular in nature. So the circular DNA can be single stranded or can be double stranded. So suppose there is a DNA, circular DNA, which is single stranded, and it has 4 bases. Okay, so 3 bases are present, that means it is single stranded. 3 base pairs are present, that means it is double stranded. 4 bases are present, that means the DNA will be single stranded. Okay, and if it has 4 base pairs, that means that will be double stranded. Okay, so suppose these are the nucleotides, and these are the phosphodiester bonds. So how many phosphodiester bonds are present? 4 phosphodiester bonds are present. Okay, so here the answer will be n. And what is the n? N is number of bases, and it is 4. So the phosphodiester bond is also 4. Okay, and what about 4 base pairs? So suppose this is single stranded circular DNA. In which these circles are representing the nucleotides. Okay, this is another strand of DNA, circular DNA. Okay, so this is double stranded circular DNA. How many phosphodiester bonds are present? 1, 2, 3, 4. 1, 2, 3, 4. So 8 phosphodiester bonds are present. So the answer will be 2n. The answer will be 2n. So I will write here. The number of phosphodiester bond is 2n. So here the number of phosphodiester bond is 2n. So the answer is 8. Okay, this is what you have to remember. Okay, and remember one thing: the nitrogenous base is always attached with the sugar, and that is why in a double strand DNA, the number of the sugar will be equals to the number of nitrogenous bases, and the number of phosphate will also be same. Okay, and the sugar and nitrogenous base, they are attached with or linked with beta and glycosidic bond, and that is why the number of beta and glycosidic bond will also be same. Okay, and that is why I will write here that point: in a double-stranded DNA, in a double-stranded DNA, in a double-stranded DNA, I will write here that the number of sugars will be equal to number of phosphates will be equal to number of nitrogenous bases. Okay, and that will be equals to the number of beta and glycosidic linkage. Glycosidic linkage in a double-stranded DNA. Okay, now, so we are done with the DNA. Now, when I talk about the length of the DNA, the length of the DNA is very large, very, very high as compared to the size of the nucleus in which it has to be accommodated, or the size of the prokaryotic cell in which it has to be accommodated. For example, in case of human being, if I measure the length of the DNA present in the 46 chromosomes, it will be about 2.2 meters. And I have to accommodate or keep those that 2.2 meter DNA in a very small space. Or if I talk about the E. coli, the length of the DNA is about 1.36 millimeter, and I have to accommodate this such a long DNA in a very, very small space in the E. coli. So how is this achieved? How such long DNA, such a huge size DNA are accommodated in such a very small place? This is done by the packaging or coiling or compacting compaction of the DNA molecules, and that packaging of the DNA requires some positively charged structures, and those structures are nothing but the proteins. In case of eukaryotes, the proteins which are positively charged and responsible for packaging of the DNA are histone proteins. But remember one thing, it is not only the histone proteins which are responsible for packaging of the DNA, some non-histone proteins are also present. I repeat, because I have to accommodate such a long DNA in a very small place, I have to pack that DNA, I have to coil that DNA molecule. But the DNA molecule being negatively charged, so when why the DNA is negatively charged? Because of the presence of phosphate. Because of presence of phosphate. Okay, so if I write a phosphate group over here like this, so in this case, in the physiological pH, what happens? In the physiological pH, the H plus will be released, or that means this OH will undergo deprotonation. So in this case, what will happen? It will be O minus and it will be O minus, not OH. And that is why the phosphate is negatively charged, and because the phosphate is present in the DNA, that is why the DNA is also negatively charged. Okay, so if this is the negatively charged DNA, so here negative charge will be there, here also negative charge. Wherever there is phosphate, there will be negative charge. So if I want to coil this DNA, then what will happen? The negatively charged will come closer to each other, and because we know similar charges repel each other, that is why this coiling will not take place. So to allow this coiling, or to cancel the interaction between the negative charge of the same DNA molecule or same strand, I have to provide some positive charge from outside, and that positive charge from outside is provided by the histone proteins. And any protein which is positively charged, that is called as basic proteins. Okay, so I will say the packaging of the DNA, why it has to be done? To accommodate long DNA in a very small place. Okay, and I have mentioned the data. Okay, in case of human it is 2.2 meter, in case of eukaryote it is 1.36 millimeter. Please note down those points, they are also mentioned in the NCERT. Okay, and so this packaging of the DNA is carried out by histone proteins. It is carried out by histone protein. So if I talk about the packaging of the DNA, it takes place in both eukaryotic cell as well as prokaryotic cell. In both, in case of eukaryotic cells, this is achieved by proteins. Okay, and the proteins which are involved in the packaging of the DNA, they are of two types. One is histone protein and second is non-histone protein. One is histone protein, second is non-histone protein. And the histone proteins, they are positively charged. And because they are positively charged, so I will say the histone proteins are basic in nature. So they are basic proteins. because they are rich with basic amino acids. Okay, what about non-histone proteins? They are, see, when the DNA is packed in the initial stage of the packaging, only the histone proteins are involved. In the later stage, non-histone proteins are involved, and the non-histone proteins are negatively charged. They are negatively charged, and because they are negatively charged, so they are called as acidic proteins. Acidic proteins, and these proteins are involved in the initial level of packaging. So when the DNA is packaged, in the initial level, the histone proteins are involved, and at the higher level of packaging, the non-histone proteins are involved. The non-histone proteins are involved, and this histone protein is of two types. One is core histone, and second is linker histone. One is core histone, and second is linker histone. So let me write that part here. Okay, so this histone protein is of two types. One is core histone, and second is linker histone. Okay, and this linker histone is H1 protein. Here H is for histone, so this H1. What about core histone? The core histone are of various types like S2A, S2B, H3, and H4. These are various histones. Okay, so what about the packaging of the DNA in case of prokaryotes? In case of prokaryotes, the histones are absent, but the DNA is packed. So who are going to pack the DNA? It is the positively charged proteins, and they are polyamines. They are polyamines, and they are positively charged. They are also positively charged, but they are not histone proteins. And remember one thing, if I talk about the prokaryote, they are classified into two groups. One is eubacteria, another is archaebacteria. One is eubacteria, another is archaebacteria. In case of eubacteria, the histone proteins are absent, whereas in archaebacteria, the histone proteins are present. So whenever the question is asked to you about prokaryote, you have to think with respect to eubacteria, because the common group is eubacteria. So while answering the questions about prokaryote, you should think about eubacteria, and that is why you will say histone proteins are absent. Clear? Very nice. So how these histone proteins, they are going to help in the packaging of the DNA. See, this green structure is histone octamer. Octamer means that will consist of 8 units, and those 8 units are nothing but the core histone molecules. That means this core octamer, it will consist of what? Two copies of S2A, 2 copies of S2B, 2 copies of H3, and 2 copies of H4. Okay, so how many proteins are present here? 8 proteins. That is why this is known as octamer. Okay, and this core histone octamer is called as new body. What do you call this one? You call this as new body. And around this new body, the DNA is wrapped. Okay, and the DNA plus core histone octamer, that is called as nucleosome. And to this nucleosome, this linker histone is present, and the exact function of this linker histone is still not known. Okay, and if you remove this core histone, linker histone, then what will happen? This DNA will not remain attached with this core histone. Okay, so here I will write one point. This is very, very important, and you will get misinformation about this, so that is why this is important. So if I talk about, if I talk about the nucleosome, so this nucleosome consists of 2 components. One is new body, and second is the DNA. And the DNA, this DNA which is wrapped around or coiled around the core octamer, that is called as core DNA. What do you call that as? Core DNA. And this new body consists of what? 8 histones. 8 histone proteins. And which histones? Core histone molecules. Okay, so in the, and this histone does not have, and this 8 histones does not include H1. And that is why I will say this nucleosome does not have H1. This nucleosome does not have H1. Okay, so I will write here, nucleosome. In the nucleosome, H1 protein is absent. H1 protein is absent. Linker histone is absent. Which histone is absent? Linker histone is absent. The linker histone is absent. That is what you have to remember. Okay, this is important. No doubt in this diagram, H1 is mentioned. That it does not mean that H1 is the component of nucleosome. No, it is not the component. Why? Because so why this structure is mentioned over here? H1 is mentioned over here because if you remove this H1, then the DNA will not remain in this form. It will slip off. That is why this H1 is present. I repeat, H1 is not a component of nucleosome. Okay, but still in this diagram, this structure is present. H1 is present because in the absence of H1, this DNA will not remain attached with this core histone. That is why it has to be present. Clear? Good. Now, now what about the packaging? Okay, so far we have learned one thing, that the DNA gets wrapped around the core histones. So the histones are involved. Okay, so suppose this is a DNA molecule, and what is the width of the DNA molecule? 2 nanometer. That means 20 angstrom. 20 angstrom. And when the DNA molecule gets wrapped around the core histone, the length will be, or the width will be, to 11 nanometer. 11 nanometer. And in this case, these structures are called as nucleosome. This structure is called as nucleosome. And in between these nucleosomes, this DNA is present, and this DNA is called as linker DNA. This DNA is called as linker DNA. Okay, so this DNA is linker DNA. This DNA is linker DNA. Okay, and this structure is one nucleosome. This structure is another nucleosome. So the nucleosomes, they give bead-like appearance. And that is why I will say nucleosomes are responsible for bead-like appearance on a string. So in this case, what is compared with string? It is the DNA. What is bead? It is the nucleosome. Okay, so this structure, so this is double-stranded DNA. The width of the double-stranded DNA is 20 angstrom, that is 2 nanometer, and the width of the nucleosome is this width. This width is 11 nanometer, so this is nucleosome. Okay, and the nucleosome will further undergo coiling, and that coil structure is called as solenoid. And the solenoid, so this is solenoid. Okay, and the solenoid has 30 nanometer width. Okay, and when it undergoes further coiling, that will be called as super solenoid. That will be called as super solenoid. And what is the width? It is 300 nanometer. Then ultimately the chromatid will be formed. Ultimately the chromatid will be formed. Okay, so here I can call this super solenoid as chromatin. I can call that as chromatin. Okay, and these are chromatids. These are chromatids. Okay, so these are chromatids. Okay, and in case of metaphasic chromosome, I have two chromatids. This is one chromatid. This one is one chromatid, and this one is another chromatid. And this one chromatid has what is the width? It is the 700 nanometer, and this one also 700 nanometer. So the entire width will be 1400 nanometer. 1400 nanometer. Clear? So this is how the DNA undergoes packing. Okay. Now, so in the lower level of packaging, it is the histone protein which is associated, but in the higher level, it is the non-histone protein. Non-histone proteins are also associated. Okay. So we are done with the packaging of the DNA. So you can see in this diagram, okay, so the DNA, these are the histone proteins, okay, and this is the core histones, okay, a core octamer, and the DNA gets wrapped around here. So this is nucleosome, this is nucleosome. Further it is undergoing coiling, and ultimately it is forming the chromosomes. Clear? Very nice. So now we will talk about the search of the genetic material. So when I talk about the search of genetic material, before that, let me drink some water. Okay, so let's talk about the search of genetic material. Previously, many scientists, they used to think that the genetic material is protein, but later on it was found that it is not the protein but DNA which is genetic material. No doubt some non-cellular organisms like viroids and RNA viruses or riboviruses, they have RNA as genetic material. But if I talk about organisms, you have to think in general about the cellular organisms. And in case of cellular organisms, the genetic material is double-stranded DNA. So I repeat, initially, when the research was not done, before that, the scientists used to think that the genetic material is protein, not the DNA. Okay, so how the scientists came to conclusion that the genetic material is DNA, not the protein? Let us try to understand that. So the first scientist who did an wonderful experiment which helped us in later on which helped us to find out the chemical nature of genetic material. It was Griffith. Griffith never said that the genetic material is DNA, but his experiment later on helped us to find out, helped the other scientists to find out the genetic material as DNA. So what did Griffith Sir did? So first thing which you have to remember is that when did he perform his experiment? It was 1928. 1928. The specimens or the organisms which was used by him in his experiment were mice and a bacterium. And the bacterium which was used by him was Streptococcus pneumoniae, or you can call that as Pneumococcus pneumoniae. And the name itself is suggesting us that this bacterium causes a disease, and the disease is pneumonia. And this bacterium has two forms. One is R form, that means rough, and another is smooth form, which means S form, that means smooth. And these forms have various strains like R1, R2, R3, R4, etc. And S also has various strains like R1, S1, S2, S3, S4, etc. And which forms were used by the great Griffith is important. Okay, so which forms were used by him? It was R2 and S3. R2 and S3. R is rough and this does not have any capsule. We know the prokaryote have envelope. The envelope consists of capsule, which is a form of glycocalyx. Okay, second is cellule, third is plasma membrane. So in this case, in R form, there is no capsule. That is why that will be called as non-capsulated. And the chemical which is responsible for pathogenicity or disease of which type of disease? Pneumonia is present in the capsule. So if the bacterium does not have capsule, it will not cause pneumonia. So that is why it is non-pathogenic or non-virulent. Whereas this S form has capsule. And because this capsule has the chemicals responsible for pneumonia, that is why it will cause disease. And hence I will call this S form as pathogenic or virulent. Okay. So how did he perform this experiment? How was his experiment? Let us try to understand. So he took live R2 and he injected that live R2 into a mice, healthy mice. And he found that there was no symptoms of pneumonia observed. No symptoms of pneumonia were observed. And that mice lived. In the second case, he injected live S3 into healthy mice and he observed the symptoms of pneumonia. Here I have not mentioned pneumonia. It is obvious we will talk about the pneumonia symptoms. And later on the mice died because of pneumonia, and he performed autopsy. And when he performed autopsy, he found that the dead mice had the live S3 form of bacteria. Then he killed that S3 by providing heat. So those bacteria will be called as heat killed S3, and that heat killed S3 was injected into the mice, healthy mice, and he found there was no symptom of pneumonia, and the mice survived. In the next case, he injected the healthy mice a mixture, and that mixture contained dead S3, and that dead S3 was heat killed S3 and live R2. And he found that healthy mice developed the symptom of pneumonia, and the mice died. When the mice died, he performed autopsy, and he observed that in that mice he observed, or he found, or he recovered live R2 and live S3. He was shocked when he found live S3. Why? Because the S3 which was injected was dead S3, killed S3, that means dead S3. But here from the mice he obtained or recovered the live S3. So to interpret this, he said, so how did he interpreted this one? He said that suppose this is heat killed S3. So from this heat killed S3, certain chemicals might have released. Okay, so suppose these are those chemicals. Okay, so in this case, he said that some chemical from this heat killed S3 was released, and those chemicals entered into live R2. So when they entered into live R2, the diagram will be like this. Okay, and these chemicals, they helped. Okay, so this is now live R2. Okay, and these live R2 were later on modified into live S3. They are modified into live S3. Okay, so this is live R2. Okay, so here I am having those chemicals. Okay, so he said that these chemicals which came from the heat killed S3, they are responsible for causing the change of the R2 into S3 form. And he called those chemicals, just a minute. Or just a minute. Let me do one thing. Let me write only one. Why? Because the chemical is basically DNA, and DNA is present once. So one DNA is present here. One DNA is present here, right? So he said some chemical was released from this heat killed S3, and that entered into live R2, and after some time this live R2 was converted into live S3, and he called this chemical as transformation principle. The chemical was called as transformation principle, and later on it was found that the chemical was nothing but DNA. It was nothing but DNA. Okay, so in this case what is happening? One form is getting converted into another form. R form is converted into the S form. That is why this process is called as transformation, and the chemical which is responsible was named by Griffith as transformation principle. So what is the drawback of his experiment? He could not say the chemical nature of this transformation principle. He could not say that whether this chemical was protein or RNA or DNA. He could not say that. So to characterize this chemical, 3 different scientists came and they performed experiment, and they were Avery, McLeod and McCarthy, and they performed this experiment in between 33 to 1944. Okay, so how did they characterize the chemical nature of this transformation principle? which was of Griffith's experiment. So they took heat killed S3 and they obtained the chemicals from the heat killed S3, and after obtaining the chemicals from the heat killed S3, they purified that. Okay, so when they purified that, that means they purified the DNA from that heat killed S3, they purified the RNA from the heat killed S3 and protein from the heat killed S3. And when they obtained the DNA from the heat killed S3, they divided that DNA into two parts. In one part only DNA was kept, in another part DNA was added with the DNase enzyme. DNase enzyme means that enzyme which causes the breakdown of the DNA. RNA was also done like that. RNA and RNA plus RNase, protein was also done like that. Okay, now suppose these are culture mediums. These are test tubes. In these test tubes, let me take some bacteria. Which type of bacteria? R2 bacteria. These are R2 bacteria. These are R2 bacteria. These are R2 bacteria. So what did they do? They injected this DNA from the heat killed S3 into this culture medium. And they found that in this case, what happened? In this case, some bacteria remain R2 form and some bacteria, they got changed into S3 form. In which form? S3 form. Okay, that means here what happened? Here the transformation took place. The transformation took place. Okay, so let me write here this part. Okay, so this is indicating S3. Okay, and this one is indicating R2. Okay, now in this case, when they injected DNA plus DNase, they found that there was no change in the form of this bacteria. That means there was no, so I can write like this. So only R2 form of bacteria were observed. That means there was no transformation. There was no transformation. Transformation was not observed. And when they injected the RNA from the heat killed S3 into the culture medium containing the R2 form, they observed that there was no transformation. That means only the R2 forms were observed. Okay, here also only R2 form, here also only R2 form, and here also only R2 form of bacteria were observed. Okay, so from here I can conclude that when I add a DNA molecule, some of the R form are getting converted into S form. Here all the R form are not getting converted. Why? Because it is not necessary that all the R form of bacteria will receive or uptake the DNA. It is not necessary. So only those R form of DNA, R form of bacteria which uptake the DNA, only those will be converted into S form. Okay, and that is why some R form and some S form were observed, and here S form is formed. That means here transformation is taking place. Okay, but in this case I added DNA, but along with DNA I added that enzyme which causes the breakdown of the DNA. That is why there was no transformation. And that is why from this I can conclude that the transformation principle of the Griffith Sir was nothing but the DNA. So this is how the chemical nature of the transformation principle or the genetic material was observed. Okay. Now, later on, Hershey and Chase, they performed a wonderful experiment in the year 1952. And from this experiment, it was concluded without any hesitation, without any doubt, unequivocally, that it is DNA which is responsible, which is the genetic material. Okay, and they took E. coli. So this is E. coli bacterium, and this is the bacteriophage. And in this bacteriophage, the genetic material is DNA, double-stranded DNA. And this bacteriophage has mainly two components. One is capsid. Second is the genetic material. The capsid is proteinaceous and the genetic material is DNA in nature. The protein contains amino acids, and one of the amino acids, and some of the amino acids like methionine and cysteine, they have sulfur. So if you provide a radioactive sulfur, then what will happen? The capsid of the protein will be labeled. The capsid of the protein will be labeled. But if you, and we know in the nucleic acid, that is the DNA, we have phosphates. So if I provide radioactive phosphorus, then the DNA will be labeled. Okay. So in one case, what did they do? In one case, they took a virus. In which the protein was labeled. That is, the capsid was labeled, and the DNA was not labeled. That means in this case, the bacteriophage was in the bacteriophage, which one was labeled? The capsid was labeled. Okay, and to label the capsid, which was used, it was the radioactive sulfur. It was S35. In another case, what did they do? They did not label the capsid. Rather, they labeled the DNA. And to label DNA, they used radioactive phosphorus, that is P32. Okay, so here the bacteriophage has labeled DNA. Here the bacteriophage has labeled capsid. And these are basically E. coli. So they allowed these bacteriophages to infect this bacteria. Okay, and they knew, Hershey and Chase, they knew one thing, that it is the genetic material of the virus which will enter into the bacteria. They had no idea whether the genetic material was DNA or RNA, DNA or protein. They had no idea. But they knew one thing: it would be the genetic material of the virus which would enter into the virus. Okay, so here you can see this is the virus. Okay, and here you can see the genetic material is entering into it. Okay, so after entering, the capsid will be empty. So the capsid gets empty, and then what did they do? They blended this one. When they blended, what will happen? This empty bacteriophage will get separated from this bacteria. So this E. coli has two components. This is DNA. This one is suppose this is the DNA of this bacterium, and suppose this is the DNA of the bacterium, and this is the DNA of the bacteriophage. Okay. Now what happened? Suppose both of these are the DNA from the bacteriophages. Both are DNA from bacteriophages. So initially they allowed the bacteriophages, that there is the viruses, to infect this bacterium. Then they blended, which helped the bacteria to get rid of the viruses. Then what did they do? They did the centrifugation. Here also they did the same thing. When I perform the centrifugation, then what will happen? Suppose this is the centrifuge tube. So in the centrifuge tube, I will get the pellet. At the bottom side, I will get the dense part, the dense substance, and which is known as pellet. And the upper side, it will be supernatant. Okay, and the supernatant, and what will be present in the supernatant in this case, and what will be present in the pellet? See, in the pellet, I said the pellet will contain those substances, structures which will have more density, higher mass. Okay, so which will have higher mass? It is the bacteria. Okay, so in this pellet, what will be present? It is the bacteria. That means E. coli will be present, or I will say bacteria will be present. Okay, for bacteria I am writing BAC. Okay, and that bacteria will have the genetic material of the virus. So I will say this pellet contains bacteria plus genetic material. GM is genetic material of virus. Okay, so what about the supernatant? The supernatant will contain the non-genetic component of the virus. It will contain non-genetic part of virus. Okay, so in the first case, they observed that see, bacteria means cells. Bacteria means kya? What cells? So in the first case, they observed that the pellet did not show any radioactivity. That means the bacterial cells, they did not show any radioactivity. And in first case, which was labeled protein. That means the protein did not enter into the cell. I repeat, the cells or the pellet, they will have the genetic material of the virus. Okay, and because in this case, in the first case, the pellet did not show any radioactivity. That means in this case, and because this in this case supernatant showed radioactivity. That means the proteins, they did not enter into the proteins of the virus did not enter into the bacterial cells. In the second case, they observed that the bacterial cells, that is the pellet, that showed the radioactivity. And in this case, which was labeled, it was the genetic DNA of the virus. Okay, and in this case, the supernatant did not show any radioactivity. That means here I can conclude it was DNA which had entered into the virus. Into the E. coli cells, bacterial cells. Okay, so from this they concluded that okay, it is DNA which is the genetic material. Okay, now let us talk about the DNA replication. So what do you understand by DNA replication? This is a process. This is a very, very complex process in which a single double-strand DNA is produces two double-strand DNA molecules. This is DNA replication. And the main enzyme which is involved in this process is Dna polymerase. But it does not mean only Dna polymerase is involved in this case. No. Several proteins, different types of proteins, different types of enzymes are involved in this case. And the DNA which undergoes replication is called as parental DNA. And the DNA which are produced are called as daughter DNA. So I will say DNA replication is that process in which one parental DNA is converted into two daughter DNA molecules. And these two daughter DNA molecules are exactly identical to the parental DNA. Okay, and this DNA replication is always semi-conservative. It is always semi-discontinuous. It can be unidirectional or it can be bidirectional. Okay, so what is semi-conservative, semi-discontinuous? I'm going to explain you. But before that, let me tell you about unidirectional or bidirectional. So in case of eukaryotes, the DNA replication is unidirectional. Whereas in case of prokaryotes or the mitochondrial DNA or the plastidial DNA, the replication is bidirectional. Unidirectional means the replication will take place in one direction. Bidirectional means the replication will take place in two directions. Okay, so what do you understand by semi-conservative? This is a method of DNA replication in which each daughter DNA molecule will contain one strand of the parent DNA. Okay, so let me write that part here. So suppose this is one double-stranded DNA. Okay, and I allowed this DNA to undergo replication. So when it underwent replication, it produced two DNA molecules. So in these two DNA molecules, I found that, so suppose this is one daughter DNA molecule. So suppose these are two daughter DNA molecules. This is one daughter DNA molecule and this is another daughter DNA molecule. Okay, so here I will say this is parental DNA, this is daughter DNA, this is another daughter DNA. So you can clearly see that in this daughter DNA, one strand is from this parent. In this daughter DNA also one strand is from this parent. And in a DNA molecule, we have two strands. Two strands means 100%. One strand means 50%, or I can say half. So I can say in this daughter, half of this parent is conserved. In this daughter, half of this parent is conserved. And this type of replication is called as semi-conservative replication. What do you understand by semi-discontinuous? See, replication is always semi-discontinuous. What do you understand by this? when the DNA molecule is synthesized, in this case, two new DNA molecules are formed, and in two DNA molecule, one is parental, one is newly synthesized. Whenever new strands are synthesized, one strand will be synthesized continuously, while the other will be synthesized discontinuously. That is why that is called as semi-discontinuous. Okay, so if both strands were synthesized continuously, the replication would have been called as continuous replication. If both strands were synthesized discontinuously, I would have called that replication as discontinuous. But here, one strand is synthesized continuously, other strand is synthesized discontinuously. That is why this is semi-discontinuous. Let me write that part here. Suppose this is replication fork. Okay, and suppose this is 3' and this is 5' and this is 3' and this is 5' and suppose this is the primer. Okay, and suppose this is the newly synthesized DNA. Okay, suppose these are primers. Okay, and okay, and here suppose these are the DNA molecules. DNA synthesized. So you can see the DNA is synthesized in fragments, but that means discontinuously. Here it is synthesized continuously, and that is why I will say that the replication is semi-discontinuous. Okay, what is replicon? Replicon is the segment of DNA which is capable of independent replication, and for replication, the sequence which must be present is origin of replication. It has to be present. Okay, and there should be some sequences which will cause the termination of replication. Okay, so the replicon is the segment of the DNA which is capable of showing or undergoing independent replication, or I can say the replicon is unit of replicon. This is unit of replicon, or I can say the smallest segment of DNA which can independently undergo replication. And this replicon is called as, it can be called as, in case of prokaryote, or specifically in case of E. coli. In case of E. coli, we call this as ORIC. Whereas in case of yeast, it is called as ARS. Where in case of yeast, not in all E. coli is a prokaryote. In all prokaryote, we do not call origin of replication as ORIC. ORIC is the origin of replication only in case of what? Sorry, just a minute. It will have here I am talking about the origin of replication. Origin of replication. Okay, so what is origin of replication? The sequence of the DNA where the replication starts. That is origin of replication. So this replicon will have the origin of replication, and that replication is the site where replication starts, and this replicon is called as ORIC in case of E. coli, ARS autonomously replicating sequence. Autonomously replicating sequence in case of yeast. Many people, they think that ORIC is the origin of replication in all organisms. No, this is the origin of replication only in case of E. coli, not all organisms. Clear? Very nice. Now, let us try to understand that how this replication is semi-conservative, is always semi-conservative was proved. It was Michelson and Stoll who performed an experiment in E. coli, and they proved that the replication is semi-conservative. And whenever the replication takes place, so suppose this is one parental DNA. So whenever the replication takes place, what happens? These two strands will separate from each other and they will act as template. Okay, now what will happen? Looking at this template, new strand will be formed. Looking at this template, new strand will be formed. Okay, so these are daughter DNA molecule. So in this daughter DNA molecule, one strand is from parent, one strand is newly synthesized. So this is semi-conservative DNA replication. So initially what did they do? They grew E. coli in a medium which was supplied with N15. Okay, and this N15 was provided in the form of NH4Cl. NH4Cl. Okay, so NH4Cl, when the E. coli was grown in a medium, that medium was supplied with NH4Cl, and that NH4Cl was uptaken by the E. coli, and that nitrogen from this NH4Cl was used to synthesize the DNA. That is the nitrogenous base. Okay, so they used the nitrogen which was supplied in the form of NH4Cl was of two types, N14 and N15. This is heavy isotope, this is light isotope. Okay, so initially they grew E. coli for several generations in which they supplied nitrogen in the form of N15 NH4Cl, N15 H4Cl. That means the DNA, the nitrogenous basis of the DNA will be completely N15. So the DNA will be N15. So I can call that as heavy DNA, N15 DNA. That means both the strands will have N15. Later on, what did they do? They allowed the bacteria to grow in a medium in which the N14 was supplied. N14 was supplied. That means they provided N14 H4Cl. Now those bacteria will use N14 to synthesize nitrogenous base, and that nitrogenous base will be used to synthesize the DNA. Okay, so in that case what will happen? So these two strands, so this strand has N15, this strand also has N15, so they get separated. So this strand and this strand, they get separated. Now the new strands are formed. So in this new strand, N14 will be used, because here you have provided the N14. Here also the N14, will be used. The N14 will be used. Okay, so here because one strand has N14, another strand has N15, that is why the DNA will be called as N15 N14. In the next step, what did they do? They allowed the same bacteria to grow in the N14 medium. That means they provided nitrogen in the form of what? N14 H4Cl. Then what will happen? The DNA strands will separate from each other, and now the DNA which will be synthesized will have N14. So this I will get. And every time they were performing the culturing of the bacteria, some bacteria they took and they performed centrifugation. Some bacteria they took and they performed centrifugation. And after centrifugation, they obtained this one. They obtained this one. They obtained this one. And later on, they compared the band. Okay, this one is the band, DNA band of the bacteria. This one is the DNA band of the bacteria. These are the DNA band of the bacteria. And later on, they compared the position of the DNA band. So if I compare the position of DNA band, then I will say that okay, in this case, the DNA will be N14-14. In this case, N15-N14. In this case, N15-N14. Why N14-N15? Because the replication is semi-conservative. That is why. So this is how they proved that replication is semi-conservative in nature. They proved that DNA replication is semi-conservative in nature. Clear? Very nice. And remember one thing, E. coli takes 20 minutes. It takes 20 minutes for division, for one round of binary fission. And here also it will take 20 minutes. That means what will be the total time? 20 plus 20, that means 40 minutes. That means it will be 40 minutes. Clear? So this is after one generation, I will get this one. After two generations, I will get this one. So if I allow the same bacteria to grow for 60 minutes, that means the 3 generations, then what will happen? All the DNA will be N14 except 2. Except 2, all the DNA will be N14. Okay, and in this case, N15 is called as hybrid DNA, N14 is called as light DNA, and N15 is called as heavy DNA. 14 is light DNA, 15 is called as heavy DNA, and 14 is called as light DNA. Okay, good. Now let us talk about the requirements of DNA replication. And before that, you must have known about the phase in which replication takes place. What is the phase? Please leave the comment. to please answer in the comment box. Okay, so the DNA replication is a discontinuous process. It does not take place in all the phases of cell cycle. It takes place in specific phase of cell cycle. What is that phase? Please mention that. Okay, so I will be eagerly waiting for your response. So for replication, I need various components. First of all, I need a template DNA or parental DNA. So I need parental DNA. Apart from that, I need DNTPs, deoxynucleoside triphosphates. For example, it will be so the DNTPs I will use. I need DATPs, DGTPs, DTTPs, and DCTPs. They are required, and the DNTPs have dual function. They have dual purpose. They have dual purpose. What are the functions? One function is that they act as substrate. They act as substrate for DNA replication. That means to synthesize DNA, this DNTP act as substrate. They undergo polymerization to form the DNA. Second, they act as energy source for polymerization. They act as energy source for polymerization. And when the replication takes place, as I said, when the replication takes place, the two strands of the parent DNA, they get separated. No doubt they do not get separated completely. Why? Because the complete separation of DNA, two strands of the DNA at a time, requires very high energy. Why? Because when I separate the DNA, that means I'm breaking the hydrogen bond present between the two strands of the DNA. Okay, and which requires huge amount of DNA, huge amount of energy, and that is why a small segment of the double strand DNA will be opened. Okay, and to open or uncoil a small segment of DNA, which will further allow the DNA replication, requires the helicase enzyme. And the helicase enzyme, what does this do? It causes the uncoiling, or this causes the uncoiling of DNA. Uncoiling of DNA. Okay, or I can say denaturation of DNA, I will say. What do you understand by denaturation of DNA? If the DNA, see, in a DNA we have two strands, and between these two strands I have hydrogen bonds, and if I break the hydrogen bonds, the strands will get separated. That is called as denaturation of DNA or uncoiling of DNA. So it causes uncoiling of DNA, and this uncoiling of DNA is also called as, what? Unwinding of DNA or denaturation of DNA. So when I perform unwinding of or denaturation of DNA, what do you understand by this? This means I'm breaking the hydrogen bond. Breaking hydrogen bonds present between nitrogenous bases of between two strands. And this breaking of hydrogen bond requires energy, and that energy is obtained from the ATP. And how this ATP will provide energy? When this ATP undergoes hydrolysis. And that is why this helicase enzyme has an activity. And which activity it will have? It will have ATPase activity. It has the ATPase activity. So because it has ATPase activity, that is why this helicase breaks or causes the hydrolysis of ATP, which will release the energy, and that released energy will be used to break the hydrogen bonds present between the two strands of the DNA. Okay, now when the DNA is denatured, what will happen? So suppose this is a double strand DNA. And if I allow the denaturation of the DNA in this area, then what will happen? In this area, okay, in this area and in this area, there will be further coiling. Extra coiling will take place. Okay, so this extra coiling, which is developed due to the uncoiling of this DNA in this region. When I perform uncoiling of this DNA in this region, what will happen? In this region, there will be more coiling, and that is known as supercoiling, overcoiling, or tension. And that tension is relieved or removed by topoisomerase. Okay, whenever the helicase opens the DNA in a certain region, what will happen in this area? In this area there will be more coiling, in this area there will be more coiling. That means I will say in this area there is tension, and the tension is removed by topoisomerase. And topoisomerase is of two types, topoisomerase type 1 and topoisomerase type 2. In case of replication, the topoisomerase type 2 is used. Which one is used? Type 2 is used. And in case of E. coli, topoisomerase type 2 is called as DNA gyrase, Gyrase. Okay, and one more thing, when you cause the unwinding of DNA, so suppose this is the DNA. So in this region the DNA is double stranded, so hydrogen bonds are present. In this region also hydrogen bonds are there, that means this is double stranded. But in this region there is no hydrogen bond. So I will say in this region the DNA has been denatured. But these two strands are complementary to each other. That means if it has A, it will have T. If it has C, it will have G. So automatically they will pair with each other. But I don't want them to pair. Why? Because if they are single strand form, then only new strand will form. That is why I have to keep them in single strand form. But in single stranded form, they are unstable. So when the helicase opens the DNA, single stranded form of DNA are formed, and those single stranded conditions are in single stranded condition the DNA is unstable. So to stabilize the single stranded form of DNA, we require certain proteins, and those proteins are called as single strand binding protein. Single strand binding protein, and they are non-enzymatic. They are non-enzymatic. Important. What about primase? See, the DNA polymerase cannot initiate replication. It will extend or it will add the DNTPs only if it has a pre-existing polynucleotide chain. And the pre-existing polynucleotide chain, that is which will have the 3' OH group, is nothing but the primer. Okay, I repeat, if suppose this is a chain, polynucleotide chain, so it will have 5' and this is, suppose this is a nucleotide chain. This is 5' and this is 3'. So at 3' I will have OH group. So if I allow the DNA polymerase to bind at this 3', then what will happen? The DNA polymerase will add the DNTP at this end, and it will cause the elongation of the DNA. But suppose there is no such nucleotide chain, and if I ask the DNA polymerase to cause the DNA polymerization, it will not perform. Why? Because to add nucleotide, the DNA polymerase should be provided a pre-existing OH group. It should be provided with pre-existing OH group. Okay, and the pre-existing OH group is provided by the primer. It is provided by the primer, and the primer is synthesized by the primase. I repeat, DNA polymerase can add a nucleotide only if it is provided with some pre-existing OH group, and the pre-existing OH group is provided by the primer. So suppose this is primer, and the primer is synthesized by the primase. Okay, and the main enzyme in case of DNA replication is DNA polymerase, and in case of E. coli, okay, see, let me write that part here. The primase synthesizes the primer. Okay, and the primer is basically made of RNA. This is basically made of RNA. Okay, and it has few nucleotides, less than 15 nucleotides. So I will call this as oligo-ribonucleotide. And this provides 3' OH group to the DNA polymerase, which will allow the DNA polymerase to cause elongation. And this is RNA. So see, this is oligo-ribonucleotide. That means this is RNA in nature. This is RNA in nature. Okay. Now what about DNA polymerase? In case of E. coli, which is a prokaryote, the DNA polymerase is of 3 types. 3 types of DNA polymerases are present. Pol means DNA polymerase. How many? 3 types are there. What are those? One is polymerase first, DNA polymerase first, second is DNA polymerase second, and third is DNA polymerase third. And among these three, which one is the main replicating enzyme, polymerase enzyme? It is the main polymerase enzyme is DNA polymerase 3. DNA polymerase 3 is the main enzyme. So what about the polymerase first? It is responsible for removing the primer and replacing with the DNA, because primer is made up of RNA. Okay, so that is why the RNA of the primer is removed and replaced by the DNA by DNA polymerase first. What about polymerase second? It is used in the repairing of the DNA. Okay. So what about ligase? If I talk about the lagging daughter strand, the lagging daughter strand is synthesized discontinuously. So that means they are synthesized in the form of fragments. So those fragments have to be joined, and in that case, which enzyme is involved? It is the ligase. And whenever there is joining of the fragments, there is formation of phosphodiester bond. So the question is here that DNA polymerase, when performs elongation or the polymerization, it also forms the phosphodiester bond. So why DNA polymerase cannot join the two strands? Because for DNA polymerase to form phosphodiester bond, it requires three phosphate groups. But in Okazaki fragments, I have only one phosphate group. That is why DNA polymerase cannot cause the joining of the Okazaki fragments. Okay, so that is why I require the ligase. What about magnesium? See, the DNA polymerase is a hollow enzyme. Okay, if it is hollow enzyme, that means it will require a cofactor, and the cofactor is magnesium. So in the absence of cofactor, DNA polymerase will be non-functional, so there will be no DNA replication. Okay, so these are the various requirements of what? DNA replication. So what about the mechanism of DNA replication? Let me explain you that part. Okay, so I'm not going to explain in detail, okay, because of lack of time. Otherwise I would have explained you in detail. Okay, so suppose this is, so whenever the helicase goes and binds to the DNA molecule, it opens up the DNA, and it will cause the formation of a Y-shaped structure, and this Y-shaped structure is called as replication fork, and the replication fork is the site of DNA replication. Okay, so let me write here, what is replication fork? This is the Y-shaped structure formed due to unwinding of DNA by helicase. Okay, so this is Y-shaped. This is Y-shaped structure. Okay, and here I will say this is the site of replication. Or site of DNA synthesis. DNA synthesis or replication. It is replication fork. Okay, so this is replication fork. Okay, this is 3' and 5', and this is 3' and this is 5'. So here, suppose this is the primer. Okay, now suppose this one is the DNA molecule which is being synthesized by DNA polymerase. Okay, suppose here this is the primer which was formed first. Okay, and once this primer is formed, what will happen? DNA will be synthesized like this, and after that, in this area, the primer will be formed by primase. Okay, and after that, DNA polymerase will come and bind, and it will elongate the DNA molecule or primer. So here, these are primers. This is primer. So here, this one is primer. Okay, and this one is DNA. This one is DNA. So this one is primer, this one primer, this one is also primer. This is 5 prime end, this is 3 prime end, 5 prime end, 3 prime end, 5 prime end, 3 prime end. Okay, so this strand, the synthesis of the daughter strands, this daughter strand and this daughter strand do not start simultaneously. The synthesis of this strand starts earlier and the synthesis of this strand start later. And because the synthesis of two strands do not start simultaneously, synthesis of one strand starts earlier, that is why the strand whose synthesis starts earlier, that is called as leading daughter strand. Leading daughter strand. I repeat, both the daughter strands, they are not synthesized, their synthesis does not start simultaneously. So the strand whose synthesis starts earlier, that is called as leading daughter strand. So in this case, this one is called as leading daughter strand. Leading daughter strand. So the leading daughter strand is synthesized continuously along the replication fork. So the replication fork moves in this direction and the daughter strand is also synthesized in this direction. Okay, and these strands are called as Okazaki fragments, and these strands are also called as what? Lagging daughter strand, because their synthesis starts later. That is why they are called as lagging daughter strands. So these are lagging daughter strands. And these fragments, so this is synthesized discontinuously, and this is synthesized discontinuously. So the fragments are called as, these fragments are called as what? These fragments are called as Okazaki fragment. So Okazaki fragment consists of what? DNA plus RNA. It consists of both DNA plus RNA. Okay, so this one is called as leading template strand. Leading template strand. Okay, so what is this? What is the polarity of leading template strand? 3 prime to 5 prime. So the polarity is 3 prime to 5 prime, and this strand is called as lagging template strand. And what about the polarity? The polarity is 5 prime to 3 prime. 5 prime to 3 prime. So it is 5 prime to 3 prime. Okay, leading daughter strand is synthesized continuously along the replication fork, whereas the lagging daughter strands are synthesized discontinuously in the form of Okazaki fragments. Why they have to be synthesized in the form of Okazaki fragment? Because DNA polymerase has only 5 prime to 3 prime polymerase activity, not 3 prime to 5 prime polymerase activity. If it had 3 prime to 5 prime polymerase activity, it would have synthesized DNA in this direction. But because the DNA polymerase does not have 3 prime to 5 prime polymerase activity, that is why that is why in this case the DNA is synthesized in this direction, that means in the form of fragments. Okay, so this was about DNA replication. Fine. Now let us talk about another point, another process, that is transcription. Which process? Transcription. Before that, let me take pause for 5 minutes. Okay, then let's talk about the transcription process. Okay, so what do you understand by transcription process? This is a process in which one double-stranded DNA produces one single-stranded RNA. Okay, and in this process, the enzyme which is involved is RNA polymerase. Which enzyme is involved here? RNA polymerase. The enzyme is RNA polymerase. Okay, and the RNA which is formed by the process of transcription, it can be messenger RNA, it can be transfer RNA, or it can be ribosomal RNA. And many students, they think that transcription is the formation of mRNA only from DNA. No, formation of rRNA, formation of tRNA, whatever RNA you are forming, from DNA, the process will be transcription. Clear? Now the RNA polymerase, which is responsible for performing transcription, in case of prokaryote is of one type. But in case of eukaryote, 3 types of RNA polymerases are there. And when I talk about the RNA, mainly the RNA are of 3 types, mainly mRNA, rRNA and tRNA. And in case of prokaryote, a single type of RNA polymerase forms or synthesizes all the 3 types of RNA. All the 3 types of RNA are synthesized by single type of RNA polymerase. But in case of eukaryote, we have 3 different types of RNA polymerase to synthesize 3 different type of RNAs. So what about RNA polymerase? First, it synthesizes, or it is responsible for synthesis of ribosomal RNA. And RNA polymerase 2, it is responsible for synthesis of mRNA. And the mRNA which is synthesized is not in the form of mature mRNA. It is synthesized in the form of pre-mRNA. And the pre-mRNA is also called as hnRNA. Heterogeneous nuclear RNA. And the RNA polymerase 3 is responsible for synthesis of what? tRNA. Okay, remember one thing, RNA polymerase synthesizes rRNA, but in the form of pre-RNA, pre-rRNA, pre-mRNA, pre-tRNA. Okay, and rRNA is of various types. And no doubt RNA polymerase is responsible for synthesis of rRNA, but this RNA polymerase 3 synthesizes 5S rRNA. So I will say rRNA except very sorry. So except 5S rRNA, all the different types of RNA are synthesized by RNA polymerase first. Okay, so what about the prokaryotic RNA polymerase? So whenever I say RNA polymerase, then you have to think with respect to prokaryotic, because if I say RNA polymerase first, that means this is from eukaryote, second from eukaryote, third from eukaryote. Okay, so the prokaryotic RNA polymerase is made up of different subunits, and the subunits are 2 alpha chains, 1 beta chain, 1 beta dash and 1 omega chain. So these are the polypeptide chains present in the RNA polymerase. Okay, so I will say the RNA polymerase, prokaryotic RNA polymerase is made of, this is made of what? 2 alpha chains, 2 beta chains, beta dash and 1 omega chain. Okay, and this RNA polymerase, which is made up of this one, is not capable of initiation of the transcription nor capable of termination of the transcription, but it causes only the elongation of the transcription. The entire process of transcription can be categorized into 3 groups: initiation, elongation and termination. And the initiation and termination of transcription cannot be performed by RNA polymerase alone. So to perform initiation of the initiation of transcription, the RNA polymerase requires the sigma factor. Okay, so when RNA polymerase binds with the sigma factor, the RNA polymerase when binds with the sigma factor, it will perform initiation of transcription. So I will say, because in the presence of sigma factor, RNA polymerase performs initiation. That is why this RNA polymerase is also called as, this sigma factor is also called as initiation factor. Why? Because this factor, in the presence of this sigma factor, RNA polymerase performs initiation of transcription. And in the presence of sigma factor, rho factor, this RNA polymerase performs the termination of transcription. Termination of transcription. And that is why we call this rho factor as termination factor. We call this factor as termination factor. Okay, so how this transcription takes place in prokaryote, let us try to understand. So I'm going to talk to you about the mechanism of transcription, but before that, let us talk about the transcription unit. So the smallest segment of DNA which is capable of independent transcription, that is called as transcription unit. Okay, so suppose, and transcription unit is basically segment of DNA. So this is segment of DNA which is transcription unit. So at one end I will have some sequence. So I am having this sequence over here. Okay, and here also I am having another sequence. Okay, so this is 5 prime, so this will be 3 prime, this is 3 prime, so this will be 5 prime, anti-parallel in nature. This is transcription unit. The RNA polymerase binds to a specific sequence of transcription unit, that is DNA, to start the transcription. And that sequence where RNA polymerase goes and binds and starts the transcription process, that is called as promoter. So this sequence is called as promoter. Okay, and this sequence where transcription ends is called as terminator. This is called as terminator. And this strand of the transcription, okay, and the sequence which is present between promoter and terminator is called as structural gene. That is called as structural gene. What do you call that as? Structural gene. Okay, and the first nucleotide of the structural gene is called as +1. Second is called as +2. Third is called as +3, like this. And the first nucleotide on the left side, that means the nucleotide just before the first structural gene is called as -1, not 0. 0 is not present. This is -2, -3, like this. Okay, and when we move from +1, so the reference is +1. So when I move from +1 to +2 or +3 like this, the movement is called as downstream, or the sequences will be called as downstream. But if I move from here to here, from +1 to -1, -2, -3 like this, this is called as upstream. So any sequence which is present here, that will be upstream sequence. Any sequence which is present here or here, that will be called as downstream sequences. Okay, so the first nucleotide present in this structural gene is +1. On the right side it will be +2, +3 like this. On the left side it will be -1, -2, -3 like this. Okay, now, so I can say on the downstream of the structural gene I have terminator. On the upstream of the structural gene I have the promoter. Okay, and promoter is the site where transcription starts, where RNA polymerase goes and bind, and terminator is the sequence where the termination of the transcription occurs. Okay, now the strand which is present, which have the 5 prime end at the promoter side, which have the 5 prime end at the promoter side, that strand is called as coding strand or non-template strand. And the strand which has 5 prime end at the terminator side, that is called as non-coding strand or the template strand. So let me write that part. So this strand is called as just a minute, non-template. So if this strand is non-template, so this strand will be template. So only one strand is used as template. Why? Because if both strands are used as template, then the genetic information transfer process will be very complex. And second, that if both strands are used as templates simultaneously, then the RNA formed from the two strands will combine with each other, which will form the double strand RNA, and which will prevent the translation. That is the formation of protein. That is why both strands are never used. Okay, only one strand is used as template. Okay, so this strand is template, this strand is non-template. So this non-template strand is also called as coding strand. Why? Because the sequence of nucleotide in the RNA is similar to the sequence of coding strand. there is one difference in coding strand. Thymine will be there. In case of thymine, in case of RNA, it will be uracil. That is the only difference. This is coding, so this strand will be non-coding. And the strand which forms hydrogen bond or pairs with the RNA molecule, that is called as antisense strand. So this strand will be antisense strand. So if this strand is antisense strand, so this strand will be a sense strand. This strand will be called as sense strand. Okay, so if I write promoter here and terminator here, if I write terminator here and if I write promoter here, then what will happen? This strand will be template and this strand will be non-template. Okay, so this is transcription unit. So what is transcription unit? This is the segment of the DNA which is capable of transcription. Okay, so this has three components: promoter, structural gene, and terminator. And the how to name these strands here? The strand which has 3' end at the promoter side, or I can say 5' at the terminator side, that is template strand. And the strand which has 5' end at the promoter side and 3' end at the terminator side is non-template strand. Okay, so this is transcription unit. So what about the mechanism of transcription? So the transcription occurs or can be explained in three subheadings. One is initiation, second is elongation, third is termination. Okay, so suppose this is one double-stranded DNA. This is one double-stranded DNA. This is one double-stranded DNA. So if I write like this, that means a number of sequences present here. Okay, so this is one double-stranded DNA. So in the first case, what is going to happen? The RNA polymerase will go and bind to the promoter. Okay, so suppose, let me write like this only to avoid confusion. Okay, so suppose this is 3' and 5' end, so this will be 3' end and this will be 5' end, right? Okay, so in the first step, what is going to happen? The RNA polymerase will go and bind to here, bind to the promoter. So RNA polymerase will go and bind to the promoter. So let me allow the binding here. So this is DNA. This is DNA transcription unit. Okay, and suppose this is the RNA polymerase. Suppose this is the RNA polymerase. This is RNA polymerase. Okay, and the RNA polymerase binds at the promoter side. So this sequence will be promoter sequence. This sequence will be promoter sequence. Okay. Now, in this next step, what is going to happen? This RNA polymerase will cause the breakdown of the hydrogen bond. So in here, what is present? Hydrogen bonds are present. Here also hydrogen bond is present. So these hydrogen bonds are broken, and to break the hydrogen bond, it will require energy, and the ATP is used to break the hydrogen bonds between the two strands at the promoter region. So this region is basically promoter region. This region is promoter region. So for promoter, I am writing P. Okay, and this is RNA polymerase. This one is RNA polymerase. Now what will happen? The hydrogen bond will break in the promoter area where the RNA polymerase is bound. Okay, so let me do that. Okay, so here I have the RNA polymerase. So this is the RNA polymerase. Okay, so do you think the RNA polymerase alone will go and bind? No, it cannot initiate. So that is why along with RNA polymerase, the sigma factor will also go and bind. Sigma factor will also go and bind. So here sigma factor is also present. So here the sigma factor is also present. Okay, now what will happen? So suppose this is +1 sequence, this is +2, and suppose this is +3, like this. These are the various sequences. Okay, now what will happen in the next step? So this is the DNA, okay, and this is the RNA polymerase. This is RNA polymerase. Okay, now suppose at the +1, this is +1, this is +2 point sequence, this is +3 sequence. Suppose at +1 I have suppose here, so here I am having sigma factor also, here I am having sigma factor also. Okay, so in this case what is happening? So at +1, suppose the sequence which is present is U is suppose T. Here suppose I am having A, and suppose here I am having C. At point 3 +3 I am having C, at +1 I am having T, so at +2 I am having suppose A. So now what is going to happen? By looking at the sequence, by looking at the nucleotide present at the +1, new nucleotide will be added by the RNA polymerase, and it will be here, because I am having A, so it will be sorry, because here I am having T, so it will be A. It will be A. So here the A will be added. A will be added. Okay. So here A is added here. So A will be formed here. And here I have A. So here U will be formed because ribonucleotide is added. So here I will have U. Okay. And phosphodiester bond will be formed between them. Okay. So this is basically 5' end, and this is basically 3' end. This is ribodinucleotide. This is ribodinucleotide. Okay, so this complex, this is double-stranded DNA, and this DNA is bound with RNA polymerase plus sigma factor, and this is called as binary complex because it has two things: RNA polymerase and DNA along with sigma factor. We cannot ignore sigma factor, no problem. So this is binary complex, and this one is also binary complex. But there is a difference. In this first case, the DNA is double-stranded. So this is called as closed binary complex. And here the DNA is opened. And this open structure is called as transcription bubble. What do you call that as? Transcription bubble. In case of replication, the unwinding of DNA forms the Y-shaped structure that is called as replication fork. Here, the structure is called as transcription bubble. And this structure is called as ternary structure. Why? Because there are three components. Ternary structure: RNA polymerase plus sigma factor 1, DNA 2, ribonucleotide. This is 3. So 3 components are there. So this is ternary structure. This is called as initiation of what? Initiation of transcription. So this is called as initiation. Initiation of transcription. Okay. Now, so when the DNA opens, or the promoter opens, at the DNA opens at the promoter region, there will be supercoiling, and the supercoiling will be removed by topoisomerase. So at this end, the topoisomerase first will go and bind. So here topoisomerase first will go and bind, and at this end topoisomerase 2 will go and bind, and they will remove the tension produced due to the opening of the DNA at this region. So this will be topoisomerase first one, and this will be topoisomerase type 2. Okay, so here I will have topoisomerase type 1, here I will have topoisomerase type 2. Now, what about the elongation phase? In the elongation phase, what about the elongation phase? So this is, in the elongation phase, what will happen? So this is the DNA molecule. This is the DNA molecule. Okay, and this is the RNA polymerase. Okay, and this one is sigma factor. This one is sigma factor. Okay, and here I have dinucleotide. So suppose this is one nucleotide, this is another nucleotide. Okay, and this is the phosphodiester bond. Phosphodiester bond. So this is dinucleotide. Dinucleotide. Now what is going to happen in the elongation phase? What will happen? The sigma factor will get released. Okay, so only the RNA polymerase will perform the elongation. Okay, so now what is going to happen? Now this RNA polymerase will move in this direction. It will move in this direction. It will move in this direction. So this strand is template strand. So this is 3' and this is 5' end. So with respect to template strand, this RNA polymerase will move from 3' end to 5' end. And when it moves, what will happen? It will cause opening of the DNA in this region, and it will add the nucleotide complementary to the nucleotide present in this template strand. Okay, so after some time you will see a condition like this. So this is RNA polymerase. This is RNA polymerase. Okay, and this one is the RNA. This one is the RNA. So this is 5' end and this is 3' end. And here in this region there will be hydrogen bond. So hydrogen bond will be there. Okay, this is 3' end, this is 5' end, this is 3' end, and this is 5' end. Okay, so this is DNA, this is the RNA polymerase, and this is the RNA molecule. This is RNA molecule. So this is elongation. This is elongation. Okay, what about termination then? What about termination? Let me explain that part here also. So the termination is of two types, but here I will talk only about the row dependent termination. Row independent termination also takes place, but here I will talk only about the row independent termination. Okay, so suppose, so here what will happen? Here a sequence will be formed. Here a sequence will be formed, and this sequence will be recognized by the RNA, so by the row factor. Which factor? Row factor. So suppose, so in this case, what is going to happen? So suppose this is the DNA molecule. Okay, this is the RNA polymerase. This is the RNA polymerase. Okay, and suppose this is the RNA molecule. RNA molecule 5' and this is 3' end. And here which bonds are present? Hydrogen bonds are present. This is 3' and this is 5' and this is 3' and this is 5' end. Okay, now what will happen? So here some sequences are produced in the RNA molecule, and that sequence will be recognized by the row factor. That will be recognized by the row factor. So suppose this is the row factor. Suppose this one is the row factor. This one is the row factor. So now this row factor will go and bind to this sequence. Okay, so this is going to bind at this sequence. So suppose this is row factor. So this row factor will go and bind to this sequence. It will go and bind to this sequence. And after binding, this row factor will move in this direction. In the mRNA, along the RNA, this row factor from 5' to 3' it will move. Okay, and the RNA polymerase will also keep on moving in this direction. So after some time, what will happen? After some time, what will happen? So suppose this is the DNA molecule. Suppose this is the RNA polymerase. Suppose this is RNA polymerase. And suppose this is the RNA molecule. Okay, and this is 5' end and this is 3' end, and here hydrogen bonds are present. And here this is the sequence. Suppose this is the sequence. Okay, this is the sequence, and suppose this is the protein row factor. So the row factor will move and it will arrive here. Row factor will move and arrive here. Okay, so this one is row factor. Which factor? Row factor. So this row factor, when it goes and binds at the transcription bubble, it will use the energy from ATP, and that energy will be used to break the hydrogen bond present between this RNA and this template DNA. So when the hydrogen bond will break, this RNA will get separated from this template strand. Okay, so when this template strand, when the RNA gets separated from the template strand, the transcription will stop. So I will say, so what is the function of this RNA row factor? It will go and bind to the transcription bubble, where it will break the hydrogen bond present between this RNA molecule and this template strand. So ultimately what will happen? Ultimately I will get this DNA molecule, double-stranded DNA molecule, okay, plus I will get the RNA molecule. So this is the RNA molecule, okay, and I will also get the RNA polymerase. So here I will get the RNA polymerase. So this is RNA, okay, and this is DNA. So this is 5' end and this is 3' end. Okay, so this is the mechanism of what? Entire process of transcription. Clear? Now let us talk about the post-transcriptional modification. And this post-transcriptional modification takes place in the eukaryote. It takes place in the eukaryote. It is not taking place in the prokaryote. It takes place only in the eukaryote. Okay. So when I talk about the post-transcriptional modification, it involves various things. What are those? Let me tell you. So those modifications which take place after transcription, that is called as post-transcriptional modification. What do you call that? Post-transcriptional modification. So what are the events? Let me tell you. So suppose this is a double-stranded DNA. Suppose this is a double-stranded DNA. Okay. So let me do one thing. So suppose this is one double-stranded DNA, and this double-stranded DNA underwent transcription. So when it underwent transcription, it will produce the RNA molecule. So suppose this is the RNA molecule. Suppose this is the RNA molecule. Suppose this is RNA molecule. Okay, and this gene is suppose split gene, and because it is a split gene, it will have intron as well as exon. So suppose this, to indicate that, let me write here, this green part is intron, and this colored part is exon. Okay, so I can say that I can call this structure as exon 1, okay, intron 1, exon 2, intron 2, and exon 3. Intron is non-coding sequence within a gene, exon is coding sequence in a gene. Okay, now so this process is transcription. Okay, and this is DNA. This is double-stranded DNA. This double-stranded DNA is nothing but the gene. This is nothing but the gene. Okay, so this will be 3' and this will be 5', and this will be 3' and this will be 5'. So this is 3' and this is 5', and this RNA is called as hnRNA or pre-mRNA. Pre-mRNA or primary transcript. You can call this as primary transcript as well. Okay, so this sequence will be corresponding to exon 1. This is intron 1. This is exon 2. This is intron 2, and this is exon 3. Now what will happen? Some modification will take place. Exon is coding sequence. So in the mature mRNA, only the coding sequence should be present. Non-coding sequence should be removed. So how does this take place? Let me tell you. Okay, so here in the first step, what is going to happen? So this is exon first, intron first, exon second, intron second, exon third, right? So in the first step, in the post-transcriptional modification, what is going to happen? At the 5' end, cap will be added. Cap will be formed, in which a nucleotide will come and bind. Which will have 3 phosphates. Okay, so already 1 phosphate is present, and GTP will come and bind, and the GTP will have methyl group. The guanosine will have methyl group. Okay, so that will come and bind. So here the cap will be formed. So here who is coming and bind? It is the GTP. So here it will have PPP. Okay, 1 phosphate which was present earlier and 2 phosphate which have come now. Okay, and actually which comes? It is the GTP. Okay, so I can say here the GTP comes and bind. Okay, and it will have methyl group. Okay, and this structure is called as cap. So I will call this component as cap, and this cap is called as 5' cap because this is present at the 5' end. Now in the next step, what is going to happen? In the next step, what is going to happen? At the 3' end, a number of adenine nucleotide will be added. Nucleotide with adenine nitrogenous base will be added. So here a number of adenine will be added. And this is called as 3' tail. Okay, this is 5' cap and this is 3' tail. Okay, so this is 5' end and this is 3' end. So this is called as capping. This process is called as capping, and this process is called as tailing. What do you call this one? As tailing. Now what is going to happen? Splicing is going to take place. Which one is going to take place? Splicing is going to take place. So in the next step, what is going to happen? The exons will be removed and the introns will be added, and this is achieved by the spliceosome complex. Which will have snRNA, small nuclear RNA plus protein. And that is why this complex is called as small nucleoriboprotein. Okay, so now what is going to happen? Now so this is the cap and these are the tails. This is the tail. Okay, and suppose this is exon 1, exon 2 and exon 3. Exon 1, exon 2 and exon 3. And this process is called as splicing. The process in which the introns are removed and the exons are added sequentially. That is called as splicing. So capping, tailing, and splicing. These are the three steps which occur in the post-transcriptional modification, and this is called as mature mRNA. This is called as mature mRNA. So this will be 5' end and this will be 3' end. This is 3' tail, this is 5' cap. And these components, 5' cap and 3' tails, they are non-coding, and they help the ribosome to translate more efficiently. Okay, and they also provide more stability to the mRNA. They help the mRNA to get transported from the nucleus to the cytoplasm. Okay, so this is post-transcriptional modification. What are the sequence of the events? It is the capping, tailing, and splicing. What is splicing? Splicing is the removal of the intron and joining of the exons in a sequence sequential manner. Clear? So this was about post-transcriptional modification with respect to what? Pre-mRNA or hnRNA. Okay, so here I can write with respect to mRNA, with respect to hnRNA or pre-mRNA, what is the sequence of the events? It is first is the 5' capping, then 3' tailing, then splicing, Okay, now let us talk about the genetic code. Okay, so the genetic code is nothing but the 3 consecutive nucleotides which code for any amino acid, which has some information, 3 consecutive nucleotides which have some informations. That is called as genetic code. So what about the features of genetic code? The genetic code are always triplet. So not triplet, always triplet. Sorry, the triplet. What do you understand by triplet? Triplet means one codon. Triplet means one codon will have 3 nucleotides, 3 consecutive nucleotides. That is why the codons are called as what? Codons are called as triplet in nature. What about second point? The codons are nearly universal. What do you understand by nearly universal? This means if I take any codon, that will code for the same amino acid in all types of organisms with few exceptions. If I take a particular codon, that will code for the same amino acid in almost all organisms with few exceptions. So what about the few exceptions? So except mitochondria and protozoa. Except these two, the genetic codes are nearly universal. They are nearly universal, and the genetic code are specific. They are specific. So when I say specific, that means genetic code are non-ambiguous. So what do you understand by specific or non-ambiguous? This means a specific codon. This means a specific codon or one codon, a given codon will code for the same amino acid. It will code for the same amino acid. For example, if I take UGG, if I take UGG, it will always code for the tryptophan. It will always code for tryptophan. It will not code for any other amino acid. So if a given codon codes for the same amino acid all the time, that is called as specificity of the codon or non-ambiguous nature of the codon. Clear? Now, this codon is degenerate. So what do you understand by codon is degenerate? If I say codon is degenerate, that means one amino acid will be coded by more than one codons. It is not necessary that one codon Okay, more than one codon. So if one amino acid code is coded by more than one codon, that is known as the degeneracy of the codon. Let me give you one example. So if I take GGG or GGC or GGA or GGU, all of these codons, they code for the glycine. They code for the glycine. Okay, so you can see one amino acid is coded by more than one codon. So this is degeneracy. And if I say here that GGG in all organisms, it always codes for glycine, and this is called as specificity. GGG always codes for glycine. This is specificity or non-ambiguity of the codon, and glycine is coded by more than one codon. That is degeneracy of codon. And the codons are non-overlapping. Codons are non-overlapping. When I say codons are non-overlapping, it means one nucleotide cannot be part of two adjacent codons. One nucleotide cannot be a component of two adjacent codons. Let me give you one example. Okay, so suppose this is one codon, okay, and suppose this is second codon. So in this case, what is I'm saying that this nucleotide is a component of this codon and this codon. So this is overlapping, and this is incorrect. They are never overlapped. They are never overlapped. What about the other point? Okay, and one more point. There are many amino acids which are coded by more than one one codon, but there are few exceptions. So what are the non-degenerate codons? Non-degenerate codons. The non-degenerate codons are UGG and AUG. AUG will always code for methionine, and UGG will always code for tryptophan. Okay, so this is non-degenerate codons. That means tryptophan is all coded by only one codon, and methionine is all coded by only one type of codon, that is AUG. Okay, now this is non-overlapping. And what about the other feature? The other feature is that the codons are commaless. The codons are commaless. That means you have to read the nucleotides continuously. That means, suppose if I write some codons here, nucleotides here like this, AUG, ACC, ACG like this. So suppose if this is one codon and this is second codon, this will be third codon. This is correct. But if I read like this, suppose AUG, ACC, ACG like this. So suppose this is first codon and suppose this is second codon. So here I am leaving this one nucleotide, and this is incorrect. I cannot leave any one nucleotide. I have to read continuously. So these are the features of what? Genetic code. Okay, and one more thing. What? See, the AUG, the codon AUG is called as start codon. It is called as start codon. Okay, and these three are called as stop codon, and these stop codons, they code for no amino acid. Okay, these are the stop codons. UUU, UCCC, these are the codons. Okay, and these codons you have to remember. Other codons you do not have to remember. These are the codons which you have to remember. Okay, now, so this is about codons, genetic code. Now let us talk about the ribosome. Okay, and before ribosome, let me tell you what is translation. Okay, what is translation? The time is 2 hours 49 minutes. Okay, my champs, I'm very sorry, it is already 2:49, 2 hours and 49, and it is 2 p.m. 2 p.m. not 2 a.m. Okay, so this is difficult to This is difficult to record the entire chapter now, and I don't want to give you the lecture of large duration, long duration. So I would do one thing. I beg your pardon for that. I do one thing, that I will not explain the DNA fingerprinting or a few parts like central dogma like that. I will try my best to cover as much as possible. In the next chapter, ecosystem, I will add few points from this chapter, and in the coming chapters I will add the remaining parts of this chapter. Okay, so never think that I'm going to skip any part. I will explain each and every part for you. Okay, trust me, trust me. So what do you understand by translation? And here you can see I have some swelling problem, so I'm not able to speak also, because whenever I'm speaking, when the tongue is touching the upper floor, I don't know what do you call that, I think palate, it is paining a lot, but still I'm trying my best. So suppose translation. So what is translation? Translation is a process in which the mRNA will produce the polypeptide, and this polypeptide can be protein or it cannot be protein. Okay, all polypeptide are not protein, so this can be protein. So in simple terms, the process in which mRNA produces protein, from mRNA the protein is produced or polypeptide is produced, that is translation. And the polypeptide is synthesized by whom? By the ribosome. So here the ribosome is used. The ribosome is the machinery which performs this translation. Okay, so if I ask you which RNA is involved in translation, what should be your answer? If your answer is mRNA only, you are incorrect. Why? Let me tell you. We know polypeptide is basically a polymer of amino acid. So when ribosome is synthesizing polypeptide, that means it is adding amino acid one after another, right? So here to this ribosome, who is going to bring those amino acids? It is the tRNA. The tRNA will carry the amino acids. The tRNA will carry the amino acids, and the tRNA will provide to the ribosome, and the ribosome will polymerize those amino acids. So tRNA is involved, and this ribosome is made up of what? So it consists of ribosomal RNA. That means 3 different types of RNA are involved. Okay, tRNA to bring the amino acid to the ribosome, RNA component of ribosome, mRNA which has informations in the form of codons. Okay, and this process is called as translation. So what do you understand by translation? Translation is the process in which ribosome synthesizes polypeptide from the informations present in the mRNA with the help of tRNA. Okay, and because this tRNA, so what is the role of tRNA and what are the components of ribosome? We should know before understanding the entire process of translation. Okay. When I talk about the ribosome, it can be 70S type or it can be 80S type, and both the ribosomes, they have 2 subunits. 70S has 50 subunits and 30 subunits. 80S has 60 and 40 subunits. So what are the rRNA which are present in these subunits? In case of 50S, the rRNA which is present is 23S rRNA and 23S rRNA and 5S rRNA. In 30S, I have 16S rRNA. In 60S, I have 28S rRNA and 5.8S rRNA and 5S rRNA. In 40S, I have 18S rRNA. So these are the different RNAs. Okay, and what are the sites of sites present in the ribosome? 3 sites are there. E is exit site. P is peptidyl tRNA binding site. A is amino acyl tRNA binding site. amino acyl tRNA binding site. Okay, so what about tRNA then? This tRNA is also called as adapter molecule. Why is it called adapter molecule? Because this transfer amino acids to the ribosome. This is why this is called as adapter molecule. And this is also known as soluble RNA. This is also known as soluble RNA. And this has structure, and it has two types of structures. It has two types of structures. So if I take the two-dimensional structure, 2D structure and 3D structure, the 2D structure is clover leaf-like structure. Clover leaf-like structure, and the 3D structure is inverted L-shaped structure. Inverted L-shaped structure. Okay, so let me draw the clover leaf-like structure. Okay, so suppose this is RNA, tRNA. So suppose this is 5' end, this is 5' end, and suppose this is 3' end. This is 3' end. Okay, and suppose these are the arms. This is the loop. This is the arm, and this is the loop. Okay. So you can see this arm is called as DHE loop. This is called as TCIC loop. And this is called as anticodon loop. And here the three nucleotides which are present are called as anticodon. This is called as anticodon. Okay, and this loop is called as anticodon loop. And this tRNA binds to the amino acid and binds to the amino acid at the 3' end. So here the amino acid will come and bind. The amino acid will come and bind at the 3' end. 3' end. Clear? So this is the structure of tRNA, clover leaf-like structure of tRNA. Okay, now what about the mechanism of translation? What about the mechanism of translation? So the translation can be explained in three headings, like initiation, elongation, and termination. The initiation consists of three phases. One is activation of standard amino acid. Second is charging of tRNA, which is also known as amino acylation of tRNA, in which amino acid is added to the tRNA. Third is formation of initiation complex. So how do they take place? Let me explain you one by one. So first of all, let me tell you what is standard amino acid. Those amino acids which are incorporated by ribosome during translation, or which are added by ribosome by translation, or I can say the amino acids which are present in the protein, they are called as standard amino acids. And how many standard amino acids are there? 20 amino acids are there, but actually it is 22. But for NEET exam, you should remember 20. You should remember 20. Clear? Now, so first of all, the standard amino acid should be activated. So for activation, energy is required, and the energy is ATP. Okay, so suppose this is standard amino acid. AA is amino acid, and this will use the ATP. That means ATP will be used by some enzyme to activate the standard amino acid, and some enzyme will be used. Some enzyme will be used. E is for enzyme. Okay, now what is going to happen? This enzyme will bind with this amino acid, and it will also bind with the ATP, and the ATP will be hydrolyzed. ATP will be hydrolyzed. Okay, so ATP will be hydrolyzed, and it will form the AMP. It will form the adenosine monophosphate. So the two phosphates, pyrophosphate, pyrophosphates will be removed. Okay, and in this case, this amino acid is inactive. But when this amino acid binds with the AMP, this is active. And I will call this as enzyme amino acid AMP complex. Enzyme amino acid AMP complex. Okay. In the next step, what is going to happen? In the next step, I am taking the same complex here. Now the same complex will bind with the tRNA. It will bind with the tRNA. And this tRNA is called as uncharged tRNA. uncharged tRNA or non-amino acylated tRNA. Now what will happen? This enzyme will transfer this amino acid to this tRNA, and it will bind at the 3' end with the help of ester bond. Okay, so here now what is going to happen? This enzyme will transfer this amino acid to this tRNA. So I will get this tRNA which is bound with the amino acid plus enzyme plus AMP. Okay, so this enzyme is adding this amino acid to this tRNA, and addition of amino acid is called as amino acylation. Okay, and this tRNA is called as amino acylated tRNA. Amino acylated or charged tRNA. Okay, and because this enzyme is also participating in the first step, the same enzyme is involved in the first two steps. Okay, and this enzyme, because this enzyme is used to form the amino acylated tRNA, that is why this enzyme is called as amino acyl tRNA synthetase. Amino acyl tRNA synthetase. So I will write here this enzyme. Here this enzyme is amino acyl tRNA synthetase. Okay, and activation of one amino acid requires 2 ATP. Why? Because AMP is produced, not ADP. That is why I will say 2 ATP are required. So I will say activation of one amino acid requires 2 ATP. 2 ATPs are required. What about the third step of the initiation? It is the formation of initiation complex. Formation of initiation complex. So how does this happen? Let me explain you. So suppose this is mRNA, and I am explaining with respect to prokaryote. So this is mRNA. Okay, so this will be 5' end and this will be 3' end. This is mRNA. Okay, this is mRNA. In the very first phase, what is going to happen? The 30S subunit will come and bind to this subunit of the ribosome will come and bind to this mRNA at the 5' end, and along with this 30S subunit, initiation factor 3 will come and bind here. Okay, so let me do that. So this is mRNA, 5' end, 3' end, and at the 5' end some sequence will be present. And the sequence is called as SD sequence. What do you call that sequence? You call that sequence as SD sequence, Shine-Dalgarno sequence, present at the 5' end of the mRNA. And now what is going to happen? The 30S subunit will come and bind here. This is the 30S subunit of ribosome. And along with that, IF3 will also get bound. So here this is the IF3. And whenever the IF3 is bound, it will prevent the binding of the larger subunit. Now what is going to happen? In the next step, what is going to happen? See, let me draw the same diagram here again. So this is the mRNA. 5' end and 3' end. Okay, and suppose this is the ribosomal subunit. Okay, IF3 will be present. And it has 3 sides. Suppose this is one side, this is second side, this is third side. 3 sides are there, and here 3 nucleotides will be there. In every side 3 nucleotides will be there. This is E side, this is P side, this is A side. So in this step now, what is going to happen? In this step now, tRNA will come and bind. And the tRNA will be charged. So it will have, suppose T is for transfer RNA, A is for amino acid. A is for amino acid. Okay, so here the transfer RNA which is bound to the amino acid will come and bind to the P side. But this is achieved along with the initiation factor 2. And initiation factor 2, when it is active, that means when it is bound to the GTP, that will cause the binding of this tRNA, charged tRNA to this P side. And this is the first tRNA, so I will call this as initiated tRNA. And this is the first amino acid, so I will call this as tRNA first. And this will be methionine. This will be methionine, formulated methionine. Okay, so here what will happen? The anticodon of this tRNA will bind with this codon at the P side, and it is bound to the first amino acid. So the first tRNA binds at the P side, In the next step, what is going to happen? In next step, initiation factor first will come and bind. Okay, and when it binds, what will happen? In that step, what will happen? The GTP which is bound. So here the GTP is also bound, or I can say IF2 GTP is bound. So now what will happen? The GTP will get hydrolyzed, it will release energy, and that energy will be used. So GDP will be produced. And now what will happen? The 50S subunit will come and bind. So here I can write that the 50S, that means larger subunit will come and bind, and at that same time what will happen? Initiation factor 3 will get released, initiation factor 2 will also get released, and initiation factor 1 will also get released. It will initially come and bind, and then it will get released. Okay, so let me draw the diagram here. This is the larger subunit. Okay, and this complex is called as initiation complex. So initiation complex has mRNA and one amino acid tRNA and ribosome. That is called as initiation complex. So this is formation of initiation complex. So what about the elongation phase then? What about elongation phase? Let me explain you that part. In elongation phase, what will happen? So suppose this is the tRNA. Suppose this is the tRNA. So suppose this is the mRNA. Okay, and this is the ribosome. These are the various sites. E site, P site, A site, and here what do I have? I have tRNA first, which is as bound with amino acid. In the next step, what is going to happen? In elongation phase, what is going to happen? Let me draw this diagram once again. E site, P site, A site, right? So this is the first tRNA bound with amino acid first. Now what will happen? Now the second tRNA bound with the second amino acid will come and bind. Okay, and for this one factor will also come and bind, and the factor is elongation factor. Elongation factor Tu will come and bind, and it requires a GTP. Okay, so the elongation factor Tu, when it is bound with GTP, that means when it is active, it will guide the binding of this second amino acid tRNA to bind at the A side. The first tRNA binds at the P side, the remaining upcoming tRNA bind at the A side. So now what will happen? Now here the second tRNA bound with second amino acid will come and bind. Okay. Now in the next step, what will happen? In the next step, there will be, let me do this, write this part again here. In the next step, what is going to happen? See, this is tRNA first, amino acid first, tRNA second, amino acid second, right? So in the next step, what is going to happen? Simultaneously, when this bond is broken at the p side, the bond between this amino acid will form. So ester bond is broken and peptide bond is formed simultaneously. The energy released during upon the breakdown of this bond will release that energy, and that energy will be used to form this peptide bond. Okay, so I can write here like this. So suppose this is the RNA. Okay, so suppose this is E side, this is P side, and this is A side. So here I have tRNA first, amino acid first, tRNA second, amino acid second. So what is happening? This bond will break, that will release energy where? At P side, and that will release energy, and that energy will be used to form a bond between these amino acids, and this bond is called as peptide bond. This bond is called as peptide bond. So where the peptide bond is formed? At the A side, Okay, so this is what is going to happen. So this will happen. So there is simultaneous breakdown, the breakdown of the ester bond between amino acid and tRNA at the P side will occur along with the formation of peptide bond between amino acid A1 and A2 at the A side. Clear? Okay, fine. Now in the next step, what is going to happen? Now next, this ribosome will move in forward direction in 3 nucleotide distance. 3 nucleotide distance it will move, and that is called as translocation. So let me write that part here. Oof. Okay, so now what is going to happen? Now this one will move in forward direction, and that is called as translocation. What do you call that as? You call that as translocation. Okay, now when this translocation requires a factor which is called as elongation factor G, which will require GTP, and because this factor is elongation factor G is involved in translocation, that is why this is called as translocase. That is called as translocation. So what is translocation? The movement of ribosome from 5' end to 3' end at by 3 nucleotide distance, 3 nucleotide long distance, that is called as translocation. Okay, so when the ribosome will move, then what will happen? This tRNA, uncharged tRNA, will move here, and this tRNA will move here. So I can write here that this will move here and this will move here. Okay, and the same steps, that is the coming of the third tRNA with third amino acid, then the breakdown of the bond and formation of peptide bond and the translocation. This will keep on this will keep on happening. This will be repeated several times. So if this is repeated several times, then what will happen? This is repeated several times, then ultimately I will get a structure like this. Okay, so suppose this is tRNA n. So amino acid n will be there, amino acid n minus 1 will be there. So like this, amino acid second, amino acid first. So this is polypeptide chain. This is Tn minus 1 and this is Tn minus 2. This is E side, P side, A side. Okay, so this is how happens. This is how happens. This is how the elongation phase take place. Okay, now what about termination? What about termination? So suppose this is tRNA. So suppose this is mRNA. Okay, and suppose this is the ribosome. And suppose these are the nucleotides. This is E side, P side, A side. Okay, E side, P side, A side. And here I am having tRNA n, amino acid n, amino acid n minus 1, like this, amino acid 2, amino acid 1. Here Tn minus 1. Okay, and here Tn minus 2. Tn minus 2 will be present, right, at the exit side. So sorry, just before the exit side. Okay, now if at the A side some stop codon is there, stop codon arrives, then what will happen? If stop codon will arrive, then no tRNA will go and bind. So in that case, which will go and bind? It is the release factor. So in that case, the release factor will go and bind. If at the A side stop codon arrives, then no tRNA will go and bind. Instead of that, release factor will go and bind. Okay, so we have three types of stop codons. We have three types of stop codons. So suppose, and release factor is also two types. I don't know what is happening. It is hanging a lot. So RF first and RF second, release factor second. So if the stop codon at the A side is UAA or UAG, then RF1 will go and bind. And if it is UAA or UGA, RF2 can go and bind. RF2 can go and bind. That means UAA, if it arrives at the A side, both RF1 and RF2 can go and bind. But if UAG arrives, only RF1 will go and bind. If UGA comes, then RF2 will go and bind. Okay. So when at the A side stop codon comes, the RF release factor, that is release factor, will go and bind at the A side. That will stop the translation. That will stop this translation. And later on, what will happen? Later, after the binding of RF, after binding of RF, what will happen? The RRF will come and bind. ribosome recycling factor. This is release factor, this is ribosome recycling factor. Along with that, elongation factor G will come and bind, which will be bound with the GTP, and IF3 will come and bind. So when this RRF will come and bind, what will happen? This will cause the dissociation of all the complexes. That means ribosomal segments will separate from each other. Okay, they will not bind with each other. Why? Because IF3 is bound here. Okay, mRNA will get separated, tRNA will get separated, polypeptide will get separated. Okay, so that will cause the termination of the completion of the translation. I repeat, if the A site is reached by a stop codon, instead of tRNA, aminosylated tRNA, RF release factor will go and bind. Okay, which release factor will go and bind? It will be decided by the nature of the stop codon. And later on, what will happen? IF3 along with elongation factor G and RRF, ribosome recycling factor, they will go and bind to this complex, which will cause the dissociation of all the components of this complex, which will cause the termination of the translation. So this is what happens in the translation process. Clear? Now, let us talk about regulation of gene expression. So what do you understand by gene expression? When a gene which is coding for protein, which has information of a protein, if it produces protein, I will call that as gene expression has completed. If a gene is responsible for forming protein, and if the protein is formed, then I will say the gene is expressed completely. The gene is expressed completely. Okay. So the gene expression can be regulated at various levels. It can be regulated at various levels. So before that, let me explain you this part. So suppose this is a double gene, which is basically a segment of double strand DNA. It will undergo, so this is nucleus and this is cytoplasm. Okay. So in the nucleus, I have DNA, and the DNA segment of DNA can be considered as gene, which will undergo transcription to produce the pre-mRNA, and the pre-mRNA will undergo post-transcriptional modification to produce the mRNA, and the mRNA will move to the cytoplasm through nuclear pore, and the mRNA will undergo translation to produce the protein. Okay, and when this protein is formed, I will say that this gene is expressed completely. Okay, and the regulation of gene expression can be controlled at 4 different levels. First level is transcription level. Second is post-transcriptional level, which will include splicing. Okay, at transcription level, I will regulate gene expression at the initiation level, and third is transport level. Fourth is translation level. So these are the 4 different levels of gene regulation. Clear? Formation of protein by this gene is called as expression of this gene, and the expression of this gene can be regulated at 4 levels. One is transcription level, and transcription level means initiation of transcription level. Okay. Second, post transcriptional modification level, which includes splicing. Third, transport level. Fourth is translation level. Okay, so these are the different levels of gene expression, regulation of gene expression. Four levels. At 4 different levels, I can regulate the gene expression. First is transcriptional level. And here also, to be specific, I will say initiation of transcription. And this is the best site for regulation of gene expression. Second is post-transcriptional modification or processing, and this includes capping, tailing, and splicing, and here I will include the splicing. Third is transport level, transport of mRNA from nucleus to cytoplasm. Fourth is translational level. Okay, and this one The best site for regulation of gene expression is the initiation of transcription. This is the best site. Okay. So to explain how this gene expression, the regulation of gene expression takes place, let me explain you the operon concept. So what do you understand by operon? Operon is the smallest DNA which is capable of expressing itself independently and which is capable of undergoing regulation by itself. So I can say operon is basically unit of gene expression and its regulation. And this concept was given by Jacob and Monod. You can note down those points. The smallest segment of DNA capable of undergoing expression and its regulation is operon. This concept was given by Jacob and Monod. Okay, and these are found only in prokaryotes. These are found in only in prokaryotes. So let me write those points. Let me check the time first. 3 hours 25 minutes. Okay. Okay, no problem. So unit of gene expression and regulation. Okay, and these are found. What about the occurrence? These are, when I talk about the prokaryotes, they are found in prokaryotes, and in the eukaryotes, EK is eukaryote, PK is prokaryote. Okay, so in eukaryote they are absent. Okay, and this concept was given by whom? Given by Jacob and Monod. So what are the various components? What about the various components? The components of an operon are promoter gene. Okay, and there will be two promoters will be there. One of the structural genes, series of structural gene, another of the regulatory gene. Okay, second is the operator gene. Third is series of structural genes. Fourth is regulatory gene. So these are the 4 different components of an operon. And this regulatory gene is indicated by I. Okay, for promoter I will write P, for operator I will write O. Okay, so suppose this is an operon. And suppose this is the promoter, this sequence is promoter, and suppose this entire sequence is the regulatory gene. Okay, or I can say this is a regulatory gene and this is the promoter of regulatory gene. And suppose here I am having another promoter. Okay, and suppose this sequence is operator gene, and here let me have some genes here. So these are genes, and these are structural genes. So suppose S is for structural gene S1, S2, S3, S4. So I will call this as series of structural genes. So this entire complex is called as what? Entire complex is called as operon. Okay, so the number of structural genes will vary from operon to operon. In lag operon, the structural genes is 3. In case of tryptophan operon, it is 5. Clear now? So this is series of structural gene. This is the promoter of series of structural gene. This is the promoter of regulatory gene, and this is the operator gene. And promoter is the site of binding of RNA polymerase, and operator is the binding site of what? Binding site of repressor. So let me write that part. So at the promoter, RNA polymerase goes and binds. Okay, and at the operator, it is the repressor which goes and binds. Okay, and this regulatory gene, when it undergoes transcription, it will produce the mRNA, and later on it will undergo translation to produce the protein, and that protein will form the regulator. It will form the regulator. And this operon is of two types. One is inducible operon, and second is repressible operon. So the operon which is normally switched off and in the presence of specific chemicals called inducer gets induced or switched on, they are called as inducible. Operon which is normally switched off, that means in which the structural genes are normally not transcribed, but in the presence of chemicals like inducer, they start undergoing transcribing, that is called as inducible operon. And the example is lag operon. And the operon in which the structural genes are normally undergoing transcription or expressing themselves, and under certain conditions or in the presence of inhibitors, I would say, they are switched off. Normally they are switched on, but in certain conditions, inhibitors, in the presence of inhibitors, they undergo switched off. They are called as repressible, which are normally switched off, and in the presence of chemical they undergo switched off. Which are normally switched off, in the presence of chemicals they undergo switched on. That is inducible. Which are normally switched on, but in the presence of chemicals they undergo switched off. They are repressible. So here, and the example is tryptophan operon. Okay, it is normally switched off in the presence of chemicals, that is inducer, it will undergo switched on. Normally switched on, in the presence of inhibitor, it will undergo switched off, that is repressible. Okay, now let us talk about the lag operon. Okay, so the lag operon is basically, it contains structural genes which are involved in the metabolism of lactose. That is why this is called as lag operon. And these genes in the lag operon ZYA, they are responsible for catabolism of the lactose. And this lag operon undergoes both positive regulation as well as negative regulation. Positive regulation is by the repressor, activator, and negative regulation is by repressor. Negative is by repressor, positive is by activator. We do not have to study about the positive regulation. We have to study about the negative regulation only. Okay, now, so there are two possibilities. Here, one case is in the absence of inducer. And in this case, what is the inducer? The inducer is the lactose. Or it can also be allolactose. So lactose or allolactose, they act as inducer. And the second case is in the presence of inducer. These are the two possibilities. So in the first case, what is happening? This regulatory gene will undergo transcription to produce the mRNA. Okay, and the mRNA will undergo translation to produce the repressor. Okay, and repressor is basically nothing but the regulator. This is regulatory gene, and the regulatory gene produces a protein which is called as regulatory protein, and regulator and this regulator is called as repressor. Let me tell you one thing. The regulatory gene produces regulator. Okay, and the regulator is of two types: positive regulator and negative regulator. The positive regulator is called as activator, and the negative regulator is called as repressor. Repressor causes negative regulation, whereas activator causes positive regulation. So here we are talking about the negative regulation of lag operon. So if it is negative regulation, obviously the regulator protein will be repressor. So what happened here? Let me repeat. The regulatory gene I undergoes transcription to produce mRNA, and that mRNA will undergo translation to produce the protein, which the protein is regulator, and the regulator is nothing but the repressor. Now this repressor will go and bind to the operator site. It will go and bind to the operator site. And here, suppose the RNA polymerase, suppose this is the RNA polymerase. The RNA polymerase will go and bind to the promoter. So this is RNA polymerase. When the RNA polymerase moves forward, transcription will take place and the gene expression will take place. But because ahead of this promoter side, this repressor is bound, that is why this bound repressor will act as an obstacle for RNA polymerase. And because of this obstacle, this RNA polymerase cannot move forward. And because this RNA polymerase cannot move forward, that is why there will be no transcription. And because there is no transcription, that is why there will be no expression of these structural genes. I repeat, the repressor produced from this gene will go and bind to the operator, and RNA polymerase will go and bind to the promoter, no problem. That means the binding of the repressor at the promoter, binding of the repressor at the operator will not prevent the binding of this RNA polymerase at the promoter, but it will prevent the forward movement of the RNA polymerase. Why? Because it will act as an obstacle. So there will be no transcription, no gene expression. Clear? But suppose there is inducer. So when this inducer, when this repressor is free, when this repressor is not bound with the inducer, it is active, and when it is active, it will go and bind to the operator. Okay. So suppose this is the mRNA and this is the repressor, and when this repressor goes and when this repressor is bound with the inducer, this repressor will become inactive. This will become inactive. Okay. So now what is going to happen? Now this inactive repressor cannot go and bind to the operator. And suppose this is one repressor. So when this repressor is bound with the inducer, this will undergo inactive. This will become inactive, and later on what will happen? When this become inactive, it will slip off this DNA. It cannot bind with the DNA. Okay, this is free repressor and this is bound repressor. This inducer can go and bind to the bound repressor or free repressor. Whenever they go and bind to the repressor, whether they are bound or free, the inducer will cause the inactivation of the repressor. Okay, and when it becomes inactive, then what will happen? This repressor will get detached from this operator. So it will get detached, so it will become free, and this cannot go and bind to this operator. Okay, so ultimately this is free now. This operator side is free now. The repressor which was bound, it got detached, and this cannot go and bind. So this is free now. Now what will happen? The RNA polymerase will go and bind to the promoter. Now this can go, this will move forward. So when this move forward, transcription will take place. So it will produce the mRNA. How many mRNA is produced? At one time, only one mRNA is produced. So this is leg mRNA. So this leg mRNA will produce 3 peptides. How many peptides? 3 peptides. So this is mRNA, and 3 peptides will be produced. Okay. So suppose 3 peptides are produced. So suppose this is one peptide, this is another peptide, and this is another peptide. So these are the 3 peptides. So these are 3 polypeptides. 3 polypeptides are there. So this polypeptide corresponds to this gene Z, and this gene will code for the beta galactosidase. Okay, and this polypeptide will form the permease, and this corresponds to the gene Y, and this transacetylase is coded by this gene A. So Z codes for beta galactosidase, Y for permease, and A for transacetylase. What is the function of beta galactosidase? It breaks down the lactose. Into galactose and glucose, and they will be used for respiration inside the E. coli. Okay, so suppose this is E. coli. If this is E. coli, Okay, and suppose this is the lactose. And if this is the, suppose this is the lactose, and this lactose is basically a disaccharide. So what is happening, what will happen? The beta galactosidase will cause the breakdown of this bond. So now what will happen? This galactose and glucose, they will become free, and they will undergo respiration to produce ATP, and that ATP will be used for the metabolism of the cell. Okay, this is one function. What about the second function? This beta galactosidase can perform isomerization of lactose to form the allolactose. So let me write that part. So lactose, beta galactosidase. Okay, so like beta galactosidase causes the breakdown of lactose into glucose plus galactose, or isomerization of lactose into allolactose. What about permease? So suppose this is the permease. Okay, this is the permease. So the permease, this is permease. Okay, and suppose these are lactose molecules, and some lactose molecules are also present here. So now what is going to happen? This permease will allow the transport of this lactose into the cell. So from higher concentration to lower concentration. So this is going to cause the facilitated diffusion of lactose from surrounding medium into the cell. What about transacetylase? It transfers the acetyl group to the galactoside. It transfers the acetyl group to the galactoside. Okay, so these are the functions of different enzymes. Okay, so this is about the negative regulation of what? Negative regulation of the lac operon. Okay, this is involved in the catabolism of the lactose. Now let me tell you the last part. Okay, before that let me tell you. So the information which is present in the DNA flows to form the protein, and the flow of genetic information from DNA to protein is called as central dogma, and the concept of central dogma was first given by Crick. Okay, so and this is the central dogma which was given by Crick is known as unidirectional central dogma. Okay, what is central dogma? The flow of genetic information is called as central dogma. Okay, it is of two types, unidirectional and reverse. Reverse is also known as teminism. Okay, so what about unidirectional central dogma? So I have DNA. From DNA, RNA is formed by transcription, and from RNA, protein is formed by translation, and DNA forms DNA by replication, and this is called as unidirectional. But in some viruses, what happens? Okay, leave about viruses. Suppose I have DNA. From DNA, RNA is formed. The process is transcription. And if from RNA, DNA is formed, that process will be called as reverse transcription. DNA to RNA transcription, RNA to protein, RNA to DNA will be reverse transcription. Transcription is performed by RNA polymerase, and reverse transcription is performed by reverse transcriptase. Okay, now this DNA will form the RNA, and the RNA will undergo translation to produce the protein, and this is called as reverse central dogma, which is also known as teminism. Okay, now what about RNA world? Initially the organisms which had developed, in those organisms RNA was acting as both genetic material as well as enzyme. Initially when RNA developed, then DNA developed. Okay, so when the RNA developed, at that time the RNA was acting both as genetic material as well as enzyme. But later on, the property of genetic material was shifted to DNA, and the property of enzyme was shifted to protein. Why? Because the criteria which are satisfied by DNA, which should be satisfied by a molecule to be genetic material, are satisfied by DNA. And genetic material should be chemically stable. It should be chemically stable, and it should replicate itself. Okay, and if I talk about DNA and RNA, which is chemically more stable, it is the DNA. Genetic material should be chemically more stable, and because RNA is less stable chemically as compared to DNA, that is why the genetic property shifted to DNA from RNA. Okay, so why DNA is more stable? Because of two main reasons. First reason is, in DNA at 2' carbon, there is no OH group. That is why this is more stable. Second reason, in case of DNA, thymine is present instead of uracil. That is why this is more stable. So because of these two reasons, because of lack of 2' OH and because of presence of thymine instead of uracil, DNA is chemically more stable, and genetic material should be chemically more stable. That is why DNA is genetic material in cellular organisms, not the RNA. What about why the properties of enzyme shifted to the proteins from RNA? Because see, RNA can act as enzymes. RNA can act as enzyme, and the example is 23S rRNA. The example is 23S rRNA. Okay, and this 23S rRNA is called as peptidyl transferase. Peptidyl transferase. Okay, why? Because this causes it is going to cause this step. So this step is performed by what? 23S rRNA. 23S rRNA. So why is it called as peptidyl transferase? Let me explain you. Okay, so suppose this is RNA. Okay, and suppose this is tRNA. Suppose this is tRNA 3, and suppose this is tRNA 4. So it will have amino acid 3, amino acid 2, amino acid 1, and this will have amino acid 4, right? And this is tRNA 2. Now what is going to happen? Now this bond will break and this peptide bond will form. So when this peptide bond is formed, that means what is happening? This peptide chain is transferred over here. This peptide chain is transferred over here. That means the diagram will look like this. This is T2, this is T3, this is T4. Okay, and here I have amino acid 4, amino acid 3, amino acid 2, and amino acid 1. So what happened? This peptide chain which was present here got transferred over here. Who is performing this one? This one. So because this RNA is transferring peptide chain, that is why this is called as transferring. That is why this is called as peptidyl transferase activity. Peptidyl transferase activity. Okay. Now, so why the property of enzyme is transferred to the proteins from RNA to enzyme? See, in all cellular organisms, the genetic property is transferred to the DNA. RNA is present, but RNA are acting as different. They have different functions. Their purpose is different. But in case of cells, RNA, DNA both are present, but only DNA is acting as genetic material. Why? Because DNA is more chemically stable. And in case of cell, RNA protein both are present, and most of the enzymes, they are proteinaceous in nature. Very few enzymes are RNA in nature, and those enzymes which are RNA, they are called as ribozyme. They are called as ribozyme. So why most of the enzymes are now proteinaceous, not RNA? Because to perform different types of reactions, different types of enzymes should be there, because enzymes are substrate specific. So to perform different types of the large number of reactions, I require large number or large types to perform different types of reactions. I require different types of enzymes. Okay, that means the enzymes should be diverse. Enzymes should be diverse. So if I compare the diversity between RNA and protein, which is more diverse, it is the protein. Why? Because in RNA, the diversity is due to the nucleotides, and the nucleotides are of 4 types: A, U, G, C. So the diversity in the RNA is due to the sequence in the sequence of the nucleotides, these 4 nucleotides. But what about the protein? In protein, I have 20 amino acids. So the difference in the protein is due to this difference in this sequence of the 20 amino acids. So which will be more diverse? This one will be more diverse. So that is why protein is more diverse. So that is why the genetic property, the enzymatic property got transferred from RNA to proteins. No doubt, even now some enzymes are RNA in nature, but most of the enzymes are proteinaceous in nature. Okay, so this is all about the RNA world. In the next class, I will talk about the DNA fingerprinting, okay, in detail, and in the upcoming chapters, I will discuss about the HGP also. Okay, so hope you understood this. Okay, so thank you so much, everyone. Thank you so much. Take care. Bye bye.